康大人做事一向周全，刚刚还在操心祠堂的布置，还特意选了。
This lecture follows on very closely with the citric acid cycle because we're going to be talking about the electron transport chain, or as I mentioned previously, oxidative phosphorylation is the alternate term. So essentially what we're going to be looking at is how the electrons that were generated and were captured in NADH or FADH2 are converted into ATP. So the key learning outcomes for today are going to be being able to describe the components of the electron transport chain, explaining the benefits of the transport chain being located within the membrane, describing how the proton motive force that is used, so this is something where you've got protons being present more on one side than the other, is converted into ATP, and also identify the determinants of the rate of cellular respiration, which remember was what we were talking about in the previous lecture. So space for questions and your lecture notes in your textbook. So, first of all, the amount of ATP that we have within our body is actually quite limited, and in contrast, our basic daily demands for energy are actually quite high. So cells are continuously recycling ADP and adding phosphate to make ATP. It's a continuous process. So this recycling uses oxidative phosphorylation, and this process occurs within the mitochondria. This is the process where ATP is formed due to the transfer of electrons from NADH or FADH2, and these both came from the citric acid cycle. These are transferred to oxygen by electron carriers. And so as I said there, these were generated by the citric acid cycle. The oxidative phosphorylation in turn generates 26 of the 30 molecules of ATP generated from the complete oxidation of one molecule of glucose. So one glucose molecule gives rise to 30 ATP, of which 26 come from the electron transport chain. And we know that the citric acid cycle also generates one, so this is where we can see the basic process. So this is the diagram that we want to talk about in terms of the overall process of oxidative phosphorylation. And again, remember, this is in the mitochondria. It's the same location that we saw the citric acid cycle occurring. So this is the membrane, this is the inner matrix. You've got oxygen coming in, converted to water, protons being pumped out. The net result is that there is a net positive charge on the outside and negative charge on the inside. Remember, this is within the mitochondria, so it's a double membrane, so it's not outside of the mitochondria, it's just outside of the inner matrix. And that charge allows protons to go through a synthase enzyme which spins, and by spinning it takes ADP and phosphate and converts it into ATP. This is the basic process. So to put some of that in context, what is the role of the mitochondria? We know that the citric acid cycle takes place in the mitochondrial matrix. The flow of electrons takes place in the mitochondrial inner membrane. So there are slight differences in location. So mitochondria have two compartments. They've got the space between the outer and the inner membrane, and this is where oxidative phosphorylation, electron transport, remember, this is where it occurs. And this is the comment that I made before, where we were saying pumping it out. It wasn't being pumped outside of the actual mitochondria, it was being pumped out from the inner versus the outer membrane within the mitochondria. And then there was the matrix, and this is bounded by that inner membrane, and this is where the citric acid cycle takes place. So here you've got an electron micrograph of a real mitochondria, and you can see these invaginations. If you zoom in, you can see this double membrane. And here is a diagram of the mitochondria. So this is the outer membrane, is the yellow region. The inner membrane is that first space. But notice with the diagram, the blue, you've got this invagination. And so this invagination gives you this inner matrix. And so this is what we're referring to. This is the inner membrane, and this is the inner matrix, which is the inside of the inner membrane space. And these folded structures are referred to as cristo, less of an issue here. But the main point of having this folding is that it massively increases the surface area and allows for greater reactions to take place within a very small organelle. So this is the main advantage of this approach. Okay, so let's get into some of the detail. Oxidative phosphorylation depends on electron transfer. That's the first point. This is very much dependent on electron transfer. Think back to the previous lecture. What were the key outputs of the citric acid cycle? It was NADH and FADH2 that were capturing protons. So, in oxidative phosphorylation, electrons from NADH and FADH2 are used to reduce molecular oxygen to water. So remember, redox, reduction, oxidation. Reduction is gain. And so what we're doing is we're taking oxygen and we're converting it to water. The oxygen is gaining the protons. And this will allow this reaction to take place. So, reaction takes place in proteins that are actually embedded in the inner membrane. They are studded within the inner membrane. And this is referred to as the electron transport chain. And they're referred to as a chain because there's a series of proteins that in essence play pass the parcel, but they're passing electrons, and by doing so, it allows it to gain and create function and also gain some potential energy. NADH, we know, is a strong reducing agent. It wants to donate that proton. So it readily donates its electrons, while we know that oxygen is an oxidizing agent and will therefore accept electrons. Oxygen, oxidizer, you can see the linkage between the two, that oxygen is an oxidizing agent. So the main force, or the driving force of oxidative phosphorylation, is the electron transfer potential of NADH and FADH2 relative to oxygen. What is the ability to transfer energy, or the potential to transfer from those molecules to oxygen? And because there is a difference, it drives it forward. Okay, let's stop there because I want to get into some of the detail about the different molecules and how they're carrying the different groups. And also, this can be a little bit of a complicated lecture, so let's break them down into small segments before we proceed. So see you in the next recording. Okay, welcome back. So, electron transport chain. As I mentioned, electron transport chain is a series of molecules. More negative the value is, the more likely it's going to occur. And as you can see, the first step, the NADH carrier. So it can be oxidized because it can lose its proton. And remember, proton and electrons, we're talking about the same thing. By passing electrons to FAD, or FMN, sorry, flavine mononucleotide, FMN, which is just another carrier. So here is FMN in the oxidized state, and here it is in the reduced state. So if we look here, we've got this nitrogen, and we've got this nitrogen gaining a proton. This is the R group, not the R group as you would see in an amino acid, but just another group that is attached, and that's why it's not shown here. And to be honest, the diagram might have been better in your textbook if they didn't detail this, but it's not a problem. Just ignore this portion. It is these two groups gain the proton. So here and here. If they gain, they can leave it, they can lose it. So again, we've got these two different states. So, actually, before I move on, structure of FMN acts as an intermediate electron carrier, which is similar to FAD. It's a similar type of approach. So the FMN is then oxidized by the next carrier, because remember, all that we're doing is we're transferring the electrons from one molecule to the next. So as soon as one becomes reduced or oxidized, it then has to repeat it to the next stage. And so the process repeats until the electron reduces molecular oxygen, which is why we need to breathe, we need oxygen. So the chain is arranged so the electrons flow to components with more positive reduction potential. So we're going from a high state to the next state, if we say a lull state, but the higher order, and that allows it to flow and for the process to occur. Now, in terms of the naming, the complexes are called NADHQ oxidoreductase. So again, you can see that this is going to be a reduction, oxidation reaction. Q cytochrome C oxidoreductase and cytochrome C oxidase. Now, what this is showing you is the free energy relative to molecular oxygen. So the more negative the value is, the more likely it's going to occur. And as you can see, the first step, the NADHQ oxidoreductase is negative, but it's not super negative. Then we've got the next one, Q cytochrome C oxidoreductase is more negative. And finally, the cytochrome C oxidase is the most negative, and therefore it's going to occur. Think of this like a hill. We're at the top of a hill. We want to go down. The electrons are just going to be able to transfer very readily because of the energy and the difference in comparison to the oxygen that we've got down the bottom. So you can see the complexes. You can see that they have prosthetic groups. So we've got iron here, we've got iron there, we've got iron again here, again and again. Notice that it is occurring over and over again. And right at the end, we've got our oxygen being converted to molecular water. But you can see electron, electron, electrons coming in, being passed, being passed, being passed. That electron gets passed and allows the ATP to be generated as that proton gradient is generated as we're going through. So, note in the diagram, NADH releases its electrons first, and we see that here. NADH releases its electron here. This is the first step, as it has a greater potential than FADH2, and we see that here. I think the easiest thing to remember here is that we've got both these molecules being generated. We've got 3 of these coming in and only 1 of those. So we have NADH being the first, and then FADH is coming through. So more protons are pumped by NADH than FADH2. And this is why differing amounts of ATP are generated depending on which carrier comes in. This has just got a greater difference. The difference from the 0 to where the oxygen is, it's greater than where the FAD is. It's slightly higher, but nonetheless, there is a difference between NADH and FADH. So this all contributes. You can see this is 1 stage lower. Therefore, it's going to give you slightly less energy through the process. I've already highlighted in the diagram, note the presence of iron in the various protein complexes. Iron is very good at shuttling between the reduced and the oxidized states, so Fe2 plus to Fe3 plus and the electron. So by shuttling backwards and forwards, it's a very good molecule. This is another reason why you need iron in your body, not just for the heme, but here, just to make ATP. Again, telling you the importance of these vitamins and minerals that are required within your body. And lastly, coenzyme Q, which is also called ubiquinone, is also an electron carrier. And we've got that here in the middle. Ubiquinone, or Q, is also an electron carrier. So it's a separate grouping to that process. For these quinones, the electron transfer reactions are coupled to proton binding and release. And remember, this is all linked to generating a proton gradient. So this should make sense why the protons are coming into play as part of this reaction. Okay, let's get into some of the detail. Now, I will go through this relatively slowly because this is complicated, but more than likely, you're going to have to go through this lecture at least twice and make sure you highlight all of the key points. So the respiratory chain consists of proton pumps. All we're trying to do is make a differential between inside versus outside of the membrane within the mitochondria, so we can have this variation in proton levels. So electrons flow across the 3 membrane complexes, which we've just seen, and by doing so, leads to the transport of protons across the inner mitochondrial matrix. That is the net result. We want to have this gradient. By having this gradient, we can use the difference in charge to drive a cellular motor, and that cellular motor is the ATP synthase, which takes ADP and phosphate and turns it into ATP. So, complex 1, NADHQ oxidoreductase. As we saw in the diagram, the previous diagram, the electron on NADH enters at complex 1, which is this NADH oxidoreductase. They flow from NADH to FMN, and then through a series of iron and sulfur clusters to ubiquinone. I have removed a lot of the content, a lot of the detail, because this is very complicated. But just understand the general flow of what is occurring here. So the flow of those two electrons from NADPH to coenzyme Q through to ubiquinone leads to the pumping of 4 hydrogen ions out of the matrix of the mitochondria. Now, I think there's a mistake there. I think that rather NADPH, that should be NADH, so please update the notes there. Go back to the leads to the pumping of 4 hydrogen ions out of the matrix of the mitochondria. Now, I think there's a mistake there. I think that rather NADPH, that should be NADH, so please update the notes there. Go back to the diagram. You can see that it is clearly NADH. Apology, that's a typo, so I'll fix that. Now, complex 2, succinate Q reductase. So remember that we had FADH2, and this was made through the oxidation of succinate to fumarate by the succinate dehydrogenase. This enzyme is actually part of complex 2. So remember, we were talking about it in the context of citric acid cycle, but you can see that it's also part of this enzymatic process. So this is going to be involved in the energy or the transfer. Now, FADH2 does not leave the complex, but its electrons bind to this iron sulfur cluster and then ultimately move to ubiquinone. There is no transport of protons in this complex. So ATP, less of it is going to be formed from the oxidation of FADH2 than NADH, because we don't have this synthetic process here. So this is one of the reasons why we've got less ATP being generated. Complex 3 was Q cytochrome C oxidoreductase. Here we've got the transfer of electrons from complexes 1 and 2 to oxidized cytochrome C, which is shown as site C. This in turn leads to protons being pumped out of the mitochondrial matrix, and for every electron, 2 protons are transported. So you can see here, this is going to be quite an efficient process. 1 electron, you're getting double the amount of protons being moved out. And lastly, complex 4, cytochrome C oxidase. You can tell that oxygen is going to be involved here. So oxidase refers to the oxygen. Here we've got the transfer of electrons from the reduced cytochrome C, which we saw up here, going to the oxygen molecule through the oxidase. So what is the net result? We've got reduced cytochrome C plus lots of protons plus oxygen, giving the oxidized form of cytochrome C. We've generated water, and also we've now pumped 4 protons between the intermembrane space. The requirement for oxygen, as I said before, is why we need to breathe. So very, very important. And we spoke about the production of CO2 in the citric acid cycle while we exhale CO2. This reaction is thermodynamically favorable. If you look here, the delta G is very large, minus 231 kilojoules per mole. So this wants to occur, as it should, because remember, we're trying to generate ATP. Not yet, but we're setting the stage for that to occur. So here's the summary diagram of the electron transport chain. We've got acetyl-CoA, we've got the citric acid cycle, we've got NADH, we've got FADH2. Remember, FADH2 is part of complex 2. So we've got complex 1 on the left-hand side, we've got 1 pumping. Complex 2, there was no pumping, so that's why we get less ATP being generated. Transfers to ubiquinone. Complex 3 gives you more protons. We've got cytochrome C as the intermediate, and then complex 4, we've got even more protons coming through. Oxygen is oxidized, and we generate water. So the net result is that we have lots of protons on the outside and fewer protons on the inner space within the matrix compared to the intermembrane space. This is all that this process is trying to do, is to try to increase this gradient. When we have a gradient, we have the potential for work through a molecular motor. Okay, let me stop there because we've now set the scene for how we're going to make use of that proton gradient. Okay, welcome back. So now we want to talk about ATP synthesis using this proton motive force. So remember in the previous part, we spoke about generating this gradient. How are we going to use that gradient to make ATP? So as we said, electron transport chain pumps protons the matrix to the intermembrane space. And this generates a proton gradient. Now the hypothesis is called the chemiosmotic hypothesis. And what this says is that the electron transport and ATP synthesis are coupled by a proton gradient across this inner mitochondrial membrane. Whenever you have a gradient, the cell has to actively work to achieve this, and also there is then a desire to go back to an equilibrium. So the cell wants to be able to move them back from one location to the next. But rather than doing so passively, it makes use of that gradient. So to reach this equilibrium, the protons do flow back into the matrix to equalize the distribution. But by doing so, excuse me, the cell undertakes some work. And so this unequal distribution is called that proton motive force, and it is a chemical gradient because you've got a difference in pH. If you've got more protons on one side, it's going to be acidic. And similarly, it's a charge gradient because you've got more positive charges on one side than you do on the other. So if you look here is an example. Location of the protons and the gradient within an individual mitochondrion. So here we've got the distribution. You can see that you've got more protons on the outside than on the inner side. You've got a high proton gradient, so this is going to be more acidic on the outside than the inner region. Protons are pumped across this membrane as the electrons flow through the respiratory chain. So this is what allows us to do this. And this gradient is going to be used as an energy potential. Now, we have a voltage difference, a membrane potential. There's going to be more charge on one side, on the other side. And this gives us a voltage difference of 0.14 volts. So the outside being more positive, and it makes sense. You've got more positive protons here, so it's going to have a slight positive charge compared to here. It's as if we've made a mini battery within the mitochondria. Think of it that way. No, actually, I've written it there. Think of it as a tiny battery. This battery, or this proton motive force, is what powers the enzyme ATP synthase. And this is the key enzyme that makes ATP, and it is a molecular rotary motor. It spins, and by spinning, it takes in ADP and phosphate, and it spits out ATP. So very, very important. Now, if we look at the ATP synthase, it is composed of a proton conducting unit and a catalytic unit. And if you think about this, this should make sense. Protons want to move from the outside to the inside, so they have to be able to pass. So this is where they're conducted or allowed to travel. And then by moving through the unit, there has to be some catalytic function. And remember, the catalytic function is basically this. It takes ADP and phosphate, joins them together to create ATP. That is the catalytic activity. So here we see it. ADP with the 3 phosphates, hydrogen phosphate, orthophosphate, and the proton gives rise to ATP with the charge and the water molecule that is also generated. Now, this complex is made up of 2 major subunits. You can tell that this is going to be a very complicated enzyme structure. So there's a complex, each made up of different subunits, and they each have multiple polypeptide chains. So we will very much look at this from a helicopter perspective. What are the key functions? There is the F0 and the F1 subunit. The F0 is embedded in the membrane. You can see that here in this diagram below. It is this blue region here. This is the F1 region, or the F0 region, sorry. F1 protrudes into the inner mitochondrial matrix, which is down here. And this is the region where the spinning takes place and also where the energy or the ATP is generated. Now, have a look at this URL. I know it's a recording, but it's in your notes too. copy that, and there is a lovely YouTube video that explains this in exquisite detail. And you can actually see it spinning and the protons moving and the ATP being generated.
Here we see an atomic model of F1FO ATP. Life requires energy. The universal biological fuel is a molecule called ATP, or adenosine triphosphate. ATP stores chemical energy in the form of a high-energy phosphate block. As the name suggests, it consists of an adenosine ring with a chain of 3 phosphate groups attached. The last of these phosphates can be split off to release the energy within the bond, resulting in adenosine the brain is having right now. The hydrolysis of ATP can be reversed by the addition of inorganic phosphate to ADP. However, this of course requires energy and is performed by a molecular power generator called ATP synthase. Here we see an atomic model of F1FO ATP synthase sitting in a membrane. This molecular motor is found in the mitochondria of eukaryotic cells and is responsible for making most of the ATP. Mitochondria, the powerhouses of the cells, turn over around 60 kilograms of ATP every day in an adult person. A long chain of biochemical processes convert the energy stored in food into a proton gradient across the inner mitochondrial membrane. These protons drive a rotation of the turbine within ATP synthase, resulting in the synthesis of ATP. Like all generators, ATP synthase consists of 2 separate motors coupled together. Within the membrane, we have the FO motor, named after the binding of antibiotic oligomycin, and at the top we have the F1 motor from factor 1. The FO motor is a proton-powered motor. It is thought that protons flow through a channel open to just the intermembrane space, where they bind to a ring of protein subunits, rotate 360 degrees, and exit through another channel exposed only to the matrix. The net flow of protons driven by the proton motor force provides the energy for the generation of rotation. The torque generated in the FO motor is transferred to the F1 motor by a central stalk or shaft. The F1 motor is responsible for generating the ATP by addition of phosphate to ADP. The top of the central stalk acts similarly to a camshaft, so that as it rotates within the F1 motor, it causes conformational changes of the catalytic subunits. The catalytic unit is made of a dimer of subunits, and there are 3 of these arranged in a ring. Catalysis occurs at the interface between the dimers. If we concentrate on one dimer, we can observe 3 distinct states. First, ADP and phosphate bind to the catalytic site. The central shaft then rotates 120 degrees to rearrange the molecules. Next, the enzyme undergoes a further 120-degree rotation, and the ADP and phosphate are fused together to create ATP. The enzyme then rotates again to return to the starting position, where ATP is released, and ADP and phosphate can be bound for the next cycle of catalysis. One key aspect of this relationship is that catalytic subunits must remain stationary with respect to a rotating central shaft. This task is performed by a scaffold on the outside of the complex, referred to as the peripheral stalk. Recent work has shown that what was once thought of as a rigid scaffold is actually dynamic and is able to accommodate changes necessary for most efficient function. Looking at this.
So, how is ATP synthesized? What are the key points? So first of all, the hydrogen ions, excuse me, these bind to the ring of the F0 subunit. It then rotates 360 degrees and releases into the matrix. So this is for every proton. In doing so, you have trapping of ADP and phosphate. Once these are trapped, they can be joined to be synthesized, and they then have to be released. And this is now the production. And then you've got another subunit, which is called the gamma subunit. This rotates in 120-degree increments, so it's going to have to rotate at least 3 times. to get back to where it started. And the dimerization of ATP synthase, so there's two of them, these can turbocharge ATP synthesis. So what you can see here is one molecule. Here is the second molecule. The spheres are the protons. Remember, they're coming in, they're going through the synthase, that is spinning, and we are taking ATP or ADP and phosphate, and we are generating ATP from this process. So, what is the energy expenditure or the energy generation? Remember, we had NADH and FADH2 coming out of the citric acid cycle, and we said that NADH is involved in the pumping of protons, so it's able to generate more ATP from its activity. And so what we see is that 2.5 ATP molecules are generated per NADH. We also said that FADH was not involved in that pumping initially as part of complex 2, so it generated less ATP, and we can see that it is 1.5 ATP molecules. This is important because we need to remember these numbers when we come to talk about beta oxidation and fatty acid degradation to make energy within the cell. So these numbers are very important. We've covered this point before. Oxidative phosphorylation is responsible for 26 out of the 30 ATP molecules for every molecule of glucose. And the availability of NAD plus and FAD, these control the rate of the citric acid cycle. Because again, these are some of those precursor molecules that are important for the citric acid cycle, and in turn this controls this pathway. So if we look here, here is the mitochondria. Oops. So here is the matrix, inner membrane space. Remember, we're looking at the inside. So where it says outside, it's still within the mitochondria. You've got the complexes, so 1, 2, 3, and 4. Remember, these are studded and present within the membrane. So the electrons coming in, they're pumping protons to the inside. Protons are on the inside, they want to exit. They exit through the ATP synthase, and by doing so, you can see the proton exits. It can get pumped back in as part of the electron transport chain, and ADP is captured by the ATP synthase and converted to ATP. And remember, there is a dimer, so there are two of these that are taking this place, or taking this reaction in place. So the top diagram is showing you the location of complexes 1 and 4 in the mitochondrial membrane. The bottom one is showing you how the citric acid products feed into the various complexes. So these are complementary diagrams. So we had acetyl-CoA coming into the citric acid cycle. Electrons were exiting either in 3 NADHs or 1 FADH2. Remember, these contribute to the different complexes. So 1, 3, and 4. We've got FADH2 being in part 2. The net result was that protons were pumped on the inside and molecular oxygen was converted to water, and we generate this proton gradient. Once those protons are inside, they go through the synthase. The synthase takes ADP and phosphate and converts it into ATP. And that is how we generate energy within the cell. So hopefully what you can see is how citric acid cycle has led to these products. These products in turn are transferring their electrons or their protons through NADH and FADH2, allowing the process to move forward. Okay, this diagram here is showing you the complete yield of oxygen or oxidation of glucose. You can see the contribution. This is showing you from glycolysis, all of the individual steps. Don't worry about, certainly do not memorize this table. This is just showing you here at the top, consumption. Then we've got generation at the various stages. And then we've got the bulk here from NADH and FADH2. Remember the 2.5 molecules per 6, so that's why there's so much. That's why the cell wants NADH, it has so much more potential. So the net result is 30 molecules in this process. Okay, let's stop there for the next lecture because I want to talk about how we can actually turn this process off through different drugs. Okay, welcome back. Now I know the previous section was relatively complicated, but remember I've tried to make it as simple as I could, but also just to highlight those key elements, so please don't get confused or caught up in the detail. Make sure you have a helicopter view right from the beginning, and then start to focus on those key aspects that I've highlighted to you. I want to finish off by talking about inhibition and an uncoupling of ATP synthesis. Now what we find is that some organisms can naturally uncouple which is really just another way of saying disconnect oxidative phosphorylation. So hibernating animals and newborns, including humans. So the uncoupling of the electron transport chain from ATP synthesis, what would be the point of this? The point of this is that we generate heat. Work is still taking place because we're going through the citric acid cycle electron transport chain, but we're not generating ATP, so the work has to give rise to heat. And this is important, particularly with a hibernating animal, for example, or a newborn, where we need to build heat and energy within the organism to maintain life and to maintain our body temperature. This is achieved by a protein called uncoupling protein 1, which should make sense. It uncouples the process. Now, how does it work? Energy of the proton gradient is normally captured by the ATPase, or the ATP synthase. Instead, it is released as heat as electrons flow through the uncoupling proton. And so what this does, and again, if we think of it like a battery, it short circuits the mitochondrial battery. It provides a loop for this to occur. Rather than being used for energy, it's just cycling through, and this generates heat. So if we look at this diagram, we've got our protons on the outside, we've got the electron transport chain actively pumping, so we've got complexes 1, 2, 3, and 4 terminating with oxygen going to water, and all of the protons being produced. And what we would normally see is that those protons get pumped through, ADP is converted to ATP. But what we have done is we have disconnected or uncoupled that reaction. So what does the cell do with all of these protons? It bypasses the synthase and goes to the uncoupling proton 1, gets pumped through, and this generates heat. And that helps to heat up the animal as a newborn or an animal that's coming out of hibernation to raise its body temperature. Really quite clever. And obviously once you get to a certain point, you turn this off and you start to make ATP. There has to be a switch between the two. Now, we can also do this through drugs, and rotenone is one example. It is found as a fish poison. It doesn't completely block ATP synthesis because electrons can still enter a complex 2, but it's going to dampen it sufficiently. It's also been implicated in, well, this process has been implicated in Parkinson's disease, and this other drug, amytal, can also be used as a sedative as part of this process. So as always, there will be some compound either naturally or something that is synthesized that can interfere. And although we haven't covered it directly, we can think of cyanide. Cyanide is also a really good example of something that can interfere with the electron transport chain and interfere with ATP generation. And why it is so toxic? Because the cells need this process, they need ATP, they need energy. You've also got antibiotics. So here we've got antimycin, which is an antibiotic from streptomyces. Here we go, I have got cyanide, azide, carbon monoxide. We know that these are all toxic. These all inhibit the electron transport chain and hence ATP synthesis, and they interact with the iron. And remember, we have the iron sulfur helping to pass the electrons down the chain. So if we block that, then we've got a serious issue taking place. So this diagram here is showing you the location of the uncouplers. So we've got the NADH going to the first complex, then to the ubiquinone. So this is where rotenone or amytal can inhibit. Antimycin can block at this location. And then right at the end, we've got cyanide, azide, carbon monoxide, right at the very last step where the oxygen is going to be involved. So there are inhibitors, there are pathways to block these steps. Now, the last thing that we want to talk about is this molecule here, dinitrophenol. This has actually been used as a weight loss agent, but it's actually quite controversial. And as we've seen with the most recent drugs that have been used as an injectable, people want to lose weight, they want to lose weight easily. So when this was found to potentially help people lose weight, it was used and exploited. Now, what this does is that it is also an uncoupler. It uncouples oxidative phosphorylation, leads to energy being lost as heat, and therefore you can lose weight. But it's toxic, okay, particularly if you have too much, because you end up having more of it being lost as heat. That excess body heat is actually bad for you, but as is the loss of energy. So this is problematic. So how does dinitrophenol work? So here is the benzene molecule, 1,2 nitro group, so dinitro and the phenol in the center. This can bind protons. That allows it to diffuse across the matrix, so at an acidic pH versus a low pH. And when it does so, it turns into this molecule, goes back and loses its protons. This disrupts the gradient. So again, by disrupting the gradient, which is what we saw here with the uncoupling protein 1, it was disrupting the gradient. The net result was heat, not ATP. That's what dinitrophenol was doing. Now you can, of course, die from this, so please don't go and source it. It's obviously not a good way to lose weight because of the side effects. Oh, okay, missing the chapter summary, but by the time you get these notes, I'll have updated that, and there will be questions. Now, this is a complex lecture, but remember, go through the recording, highlight those key points that I've covered to you, make sure you focus on those. In particular, with this one that is complicated, I'm going to be looking more for the general understanding of the principles, the key molecules, the key processes, etc., as opposed to the fine detail, just because it is so complicated. And really, our main point is, how do we link citric acid cycle? How do we link it to ATP? That is the take-home message for this part of the lecture. Okay, thank you, and see you at the next recording. Okay, welcome to lecture 14. In this lecture, we're going to cover a new pathway which is called the pentose phosphate pathway, or PPP. And here we're going to look at the importance of NADPH, its generation, and its use within the cell. So really quite important, particularly when we come to talk about oxidative stress. So the study aims for this lecture are, first of all, to understand the two products that are generated from the pentose phosphate pathway. You need to be able to describe the role of NADPH in the cell, and understand the pentose phosphate pathway and the key regulatory enzyme, or the key regulatory step. Understand what oxidative stress is and the role that glutathione plays in this process. And then we're going to talk about this molecule here, pamequin, it's also called primaquine, how this works, and then we'll also talk about glucose 6-phosphate deficiency and how it can lead to disease. So you've got your section here for the lecture notes in the textbook. So if we start off with our introduction, we know that to date, so far we've been focusing on the breaking down of glucose for energy and glycogen for energy storage. That's been all of the lecture content. But what you need to appreciate is that there is another fate for glucose 6-phosphate. And this one is it's acting as a substrate for the pentose phosphate pathway, or the PPP. Now, why is this important? This is the pathway that catalyzes the interconversion of 3 and 6 carbon intermediates, which are end results of glycolysis, with 5 carbon carbohydrates. And these are important steps. These conversions are important because they allow for the generation of pentose sugars. So remember, this is the pentose phosphate pathway, so we're generating a pentose sugar. Pentose refers to 5. These sugars are required for RNA and DNA synthesis, as well as metabolism of 5 carbon sugars that are originate from the diet. But really the key point is this one here. We'll talk about RNA and DNA in a later stage, but for this lecture, pentose phosphate pathway is an important source of the carrier, NADPH. Now we have covered NADH, and we've seen NADPH. Let's just talk about these separately. So NADH is oxidized by the respiratory chain to generate ATP. And we saw that in the previous lecture where it was fed in. Depending on where it came into the pathway, we generated different amounts of ATP. NADPH serves as a reductant, so it's a reducing agent for biosynthetic processes. So this is where we want to synthesize something within the cell. But importantly, and it protects against oxidative stress, but it is via this molecule called glutathione. So this is an important distinction to make. So here we're saying that it protects against oxidative stress, but the actual oxidative stress is dealt with via glutathione, but NADPH has a critical role in maintaining glutathione in its active state. So that's a very easy mistake and a common mistake for students to make. We're saying that it protects against oxidative stress, but the actual oxidative stress is dealt with via glutathione, but NADPH has a critical role in maintaining glutathione in its active state. So that's a very easy mistake and a common mistake for students to make. Now, when we look at the pathway, there are 2 phases, and basically this leads to the generation of NADPH, which is our key molecule, and as we mentioned above, 5 carbon sugars. Now, in plants, we're not really going to focus too much, but we know that NADPH is produced by photosynthesis. What about for other organisms? As it shows you again in bold, important step here, NADPH is needed for reductive biosynthesis in all organisms. So that's an important point, part of the synthetic process. And it is also a host of biochemical reactions. And you'll probably already predict this based on what you know about NADH. NADPH is very similar. Now, if we look at the biosynthesis steps, so these are if you look at this table, examples of pathways that require NADPH. And as it says here, the red oval will be the focus of this lecture, so this is what we're going to be looking at. But just look here. We've got fatty acid biosynthesis. We've got cholesterol biosynthesis. We've got neurotransmitter and nucleotide biosynthesis. This is all part of that biosynthetic pathway, synthesis of these molecules. Clearly, this is going to be very important. There's also the detoxification. If we go to the second one first, cytochrome P450, this is involved in dealing with compounds by the liver. So these are a series of enzymes that basically break down things that are internalized or ingested by the host, such as drugs, things that need to be metabolized and cleared. But the one that we're going to be looking at is the reduction of oxidized glutathione. So remember, glutathione deals with oxidative stress, and it is oxidized, and therefore the opposite of oxidation is reduction. This is where the NADPH comes into play. It allows for the oxidized glutathione to exist in two different states. That is an important concept, an important idea. So if we look at the pathway, as we mentioned above, two phases. One is the oxidative generation of NADPH, and the other one is the non-oxidative interconversion of sugars. This is really where we're going to be interested in this lecture. We'll talk about both, but this is going to be the focus. So in the first phase, we've got our old friend glucose 6-phosphate. It's oxidized, but it's oxidized to ribulose 5-phosphate. You might think about that molecule and think, I've heard of something which is similar. Ribose. So remember, ribose for RNA, deoxyribose for DNA. You can see that this is going to be a precursor. And I've got that on the next line. Ribulose 5-phosphate is then converted to one molecule of ribose 5-phosphate, which is a component of RNA, DNA, ATP, NADH, FAD, and coenzyme A. Super critical component of not only nucleic acids, but you can see all of these other molecules. And when you look at the structure of these other molecules, you will have seen this 5-ring structure. So you can see the overlap. But importantly here, 2 molecules of NADPH are generated. So we're able to regenerate these molecules. And what's the role of the NADPH? Well, it's that oxidative effect that it can have with the glutathione. So the reaction is very simple. As we can see here, glucose 6-phosphate, here is our phosphate molecule on the 6th position as always. We want to remove protons, so we're going to use glucose 6-phosphate dehydrogenase. NADP plus goes to NADPH and CO2, so it's also a decarboxylation type reaction. So, as it says here, phase 1, for each molecule of glucose 6-phosphate, 1 molecule of ribose 5-phosphate and 2 molecules of NADPH are generated. This is going to be the rate limiting step, because this is the one that controls the generation of the molecule that we are interested in. So this will always be the point of control. And if you look here, the Km for NADP plus is 1,000 times greater than for NAD plus. So this is the preferred substrate. And that is important because we need a source of NADPH within the cell. Now, what about that conversion? Well, remember, it's just an isomerization. So ribulose 5-phosphate goes to ribose. We've got just a reorganization of where the molecules are slightly change in shape, or slight change in shape, hence the isomerase. So we've got the same thing, but we've now generated the Km for NADP plus is 1,000 times greater than for NAD plus. So this is the preferred substrate. And that is important because we need a source of NADPH within the cell. Now, what about that conversion? Well, remember, it's just an important because we need NAD plus. So this is the preferred substrate. And that is important because we need a source of NADPH within the cell. Now, what about that conversion? Well, remember, it's just an isomerization. So ribulose 5-phosphate goes to ribose. We've got just a reorganization of where the molecules are slightly change in shape, or slight change in shape, hence the isomerase. So we've got the same thing, but we've now generated this ribose 5-phosphate, which, remember, went to RNA, DNA, ATP, etc., etc. So super, super critical molecule within the body. Now, what about phase 2? What is the fate of the ribose 5-phosphate in cells? But not only, we know what the fate is originally, because we're saying, well, it's going to be a substrate or a precursor for here. But what happens when it's in excess to biochemical needs, or we make too much of it? So ribose 5-phosphate is converted into glyceraldehyde 3-phosphate, and fructose 6-phosphate by transketolase or transaldolase. So what does this mean? We've heard of these two enzymes before. These enzymes create a reversible link between the pentose phosphate pathway and glycolysis. So if we have excess ribose 5-phosphate, it can be converted into glycolytic intermediates. So the forward reaction and the reverse reaction can occur here. So this is an important idea. Now, when we look at these tissues with an active pentose phosphate pathway, you can see the normal tissues, but let's focus just on the last one, red blood cells. And if we look here, these are all synthesis. So why am I going to the last one? Because this is the only one where there is no synthetic process, because you know that the red blood cell is there at its terminal stage, but the role is the maintenance of reduced glutathione. And this is what we are going to focus on. Don't lose sight of these above, but this will always be the main focus of this lecture, is the role of glutathione and the role of NADPH in its conversion. Okay, I'll stop there because I want to move on to talk about something else with oxidative stress. So I'll see you in the next recording. Okay, welcome back. So we finished off by talking about the focus being on red blood cells, and now we're going to move on to the key enzyme glucose phosphate dehydrogenase and oxidative stress. So what is oxidative stress, first of all? Now, reactive oxygen species, or ROS, these are normal byproducts of oxidative metabolism. You know that oxygen is involved in metabolism. We've covered this at length. It carries the electrons as part of the process. But when you use oxygen, you can also generate these reactive oxygen intermediates known as these ROS products. And these molecules can cause damage to all classes of macromolecules if they are not dealt with. So you can have a singlet oxygen, you can have hydroxyl groups, you can have hydrogen peroxide, you can have hypochlorite being generated. All of these are very, very chemically reactive and they must be dealt with. Otherwise, you start to have damage to proteins, lipid, carbohydrate, nucleic acids. So how do we deal with these products? This is where glutathione comes into play. Cells use reduced glutathione. So important, it has to be in the reduced state. And they use it as the reducing agent. So what does that mean? Well, if it is the reducing agent, it undergoes oxidation, and it undergoes oxidation in place of the vital cellular component. So instead of your nucleic acid, this takes the oxidative load. So glutathione, which is also shown as GSH, combats oxidative stress by reducing reactive oxygen species to water, to non-reactive byproducts, making them safe. Now, what is glutathione? It's just a tripeptide. It is 3 amino acids, and the terminal one has a free sulfur-hydryl group, an SH group. And so you know that this is going to be a cysteine molecule. A cysteine is the only amino acid with an SH group. Why is the SH group important? The SH group allows one molecule of glutathione to bond to another sulfur-hydryl group, forming what is called a disulfide bond. And so because we've got two joined together, this is referred to as a dimer. Now here is the structure of glutathione, and this is shown in the reduced state. So remember, it's ready to be oxidized by, let's just say, hypochlorite or hydrogen peroxide, both reactive intermediates. So here are glycine, cysteine, and glutamate. You can see the sulfur-hydryl group that is here, and you can also then imagine another group that comes along, and we can form the dimeric state. And notice we've got the same directionality that we spoke about previously, carboxyl groups and amine groups on the molecule itself. Now, a little bit of a history lesson. If we now go back to 1926, there was an anti-malarial drug called Pamoquin or Primaquin, so these are used interchangeably. People that were given this drug, in certain cases, they were found to undergo hemolysis in these patients, and this is lysis of red blood cells. So this is where they have a breakdown of the red blood cells that's not due to the malaria, but it's actually due to the drug, and this is obviously quite a damaging side effect and could actually cause death to these individuals who are already sick. It took another 30 years to figure out what was going on, and what was identified was recognized that the cause of the drug-induced hemolysis was actually the deficiency of glucose 6-phosphate dehydrogenase. Okay, so this is that key enzyme. Remember, this is the key enzyme, rate limiting of NADPH production. So that's an important point. So this is important. But why? As I've said here, why is this important? The pentose phosphate pathway is the only source of NADPH in red blood cells. Remember, we keep focusing on red blood cells. That's one where it's not used for synthesis, but it's only used to deal with oxidative stress and glutathione. So, if it's the only source, these cells are going to be dependent on its production, and this therefore requires a functional glucose 6-phosphate dehydrogenase enzyme. Now, functional or a present enzyme. So I've said here a deficiency. Now, that could either be an enzyme that is non-functional, or it may just not be present, it's expressed at very low levels. So there is a problem here. But why is this problematic? Why do the red blood cells need this functional glucose 6-phosphate dehydrogenase? The major role of NADPH in red blood cells is to reduce the disulfide form of oxidized glutathione to the sulfurhydryl form. And it is written this way, GSSG. This is showing you a disulfide bond. That's why it's shown in that particular configuration. Here, the GSH, this is shown as the monomeric form. So it exists in these two alternate states. So what we see, similar to what I just said, we have two monomers in the reduced form. We have our reactive oxygen species. These come together, they're oxidized. And remember, we've got this free proton on one molecule, but we've got two of them, so there's two protons. This leads to water being generated, and whatever has been oxidized is converted to an alcohol, or a much less reactive molecule. But then what happens? Is this the terminal reaction? Well, no, it's not. If we now look to the second step, and this is really the important part, the oxidized form interacts with NADPH. The NADPH is going to donate its protons back to the oxidized form, and we regenerate the monomer. The NADPH goes back to NADP+, and this now needs to be regenerated to make NADPH, and the cycle continues. So the net result is that we have regenerated the reduced glutathione and the monomeric form. And so why is that important? Because this is going to be present, the reactive oxygen species. And so if there is more reactive oxygen species than NADPH, this is going to start to attack different tissues. And if we're looking at the red blood cell, it's going to attack the hemoglobin, and this is what leads to the hemolysis. So as it says here, NADPH is thus essential for this reaction. You can see it here. It is critical for that interconversion. If this is not present, then we just go into the forward reaction and we just end up in the oxidized state. And then the excess reactive oxygen species are not dealt with. It's a very, very damaging process. Okay, so just finish off on this example here. Here is our oxidized glutathione. Remember I said GSSG. This is showing you the sulfurhydryl group here, the double bond. Well, not double bond, sorry, the two sulfurs that are joined together. The NADPH through a glutathione reductase. So remember, this is going to reduce the glutathione, and the protons are going to be donated. We end up with two of these because these are split down the middle, and we regenerate this sulfurhydryl group and our NADP+. So you can see that this is able to exist in the reduced versus oxidized state. And this is important for dealing with our reactive oxygen species. Okay, I'll stop there. We'll come back to disease and see why this is problematic. Okay, welcome back. So, what's the mechanism and how does this link to disease? Well, we know that glutathione, so the reduced form, is essential for maintaining the normal structure of the red blood cells because these are dependent on NADPH. If you've got cells with low GSH or glutathione levels, they're now going to be susceptible to drug-induced hemolysis. You might be going, well, okay, but why is that the case? Because there is limited levels of reduced GSH to deal with reactive oxygen species. That's really the crux of the matter. So, okay, well that makes sense. So how does this link with disease? Well, when this was first being identified, we now know that up to 11% of African Americans actually have a deficiency in glucose 6-phosphate dehydrogenase. It's an inherited characteristic for this group of individuals. Now, is this problematic? Well, in the absence of oxidative stress, people that have a deficiency in glucose 6-phosphate dehydrogenase are normal. There's no effects, there's no biological consequence, there's no hemolysis, so it doesn't present. But what we see is that when Pamoquin or Primaquin, remember the same name, just two different things, two different names for the same thing, this in turn leads to very high levels of oxidative stress. It stimulates oxidative stress. Now, notice this bit that is in yellow. I've highlighted this because this is a very common mistake. Students will think that Pamoquin or Primaquin is an inhibitor of glucose 6-phosphate. It is not an inhibitor. It is a molecule that was used to treat malaria, and one of the consequences dealing with malaria was this oxidative stress. Do not make this mistake in logic and understanding. It is not an inhibitor. Make this very, very clear in your understanding. So, how does this lead to disease? So, if we have someone that has this deficiency, NADPH is not going to be generated because remember, glucose 6-phosphate dehydrogenase was the control point and the rate-limiting enzyme, and we've got a high level of oxidative stress. All of the monomeric GSH is going to be oxidized by the reactive oxygen species. We don't have NADPH present, and so we can't return it back to its reduced state. But the level of oxidative stress, the reactive oxygen species, continues to go up because of the drug treatment. Now, reactive oxygen species are being produced. They have to interact, they're reactive, so what are they going to do? They're going to attack the most common protein in the red blood cell, which is going to be the hemoglobin. And by doing so, it damages the cell, damages the protein. Now, red blood cells also lack mitochondria, so there's no other source of NADPH. They're in trouble. They're dependent on this single source. And so what this leads to is lysis, which is what we saw above, the hemolysis that occurs. Now, you might be thinking, well, why did this population of individuals, why do 11% of people have a deficiency in glucose 6-phosphate dehydrogenase? It's actually...
这样一个惊世骇俗的计划，不是为了杀人，只是不想这本记忆失传。我真爱痴迷于这项记忆，不希望它沾染分毫的不良之念。但是爹，我没有其他选择了。现在它在我手里。
Bond comes from ATP hydrolysis, so this is an energy requiring process. There's expenditure of energy. And as I mentioned before, you need at least 4 ubiquitin molecules for the protein to be degraded in the proteasome, which is the organelle that undertakes this process. So it's not just a single addition, but actually multiple rounds. So you can see that there's going to be a lot of ATP hydrolysis for this to occur. Now, the process can be a little bit complicated, but I'll try to keep it fairly simple for you in these diagrams below. If you look here, step 1, you have a ubiquitin activating enzyme, which is just called E1, and this adenylates the ubiquitin. And what this means is that it puts the adenosine monophosphate, or AMP, onto the group. So here is the ubiquitin with a carboxyl group, ATP is expended, and we're removing pyrophosphate, which is the 2 phosphate molecules. And so this is the enzyme that does that. And by doing so, we've got the AMP that is attached. This is going to be the point of reactivity. We then have the enzyme in the second step. Enzyme 1 removes the AMP, or it actually joins together. So what you can see here is E1 is the enzyme, and the ubiquitin is physically attached. And so we can see this bond that has occurred, and we've now lost the AMP. So we could already predict that this is going to be a cysteine residue because of the sulfur hydrogen group. So as it says here in step 2, ubiquitin is then transferred onto a cysteine residue of the activating enzyme. And this is the process that we have just seen. So that was the first step, is the activation. The second one is showing the same diagram, but is now showing you that activation process. And then the third step is the ubiquitin is transferred to a cysteine residue in that conjugating enzyme. So we've got different enzymes, E1 and E2. This is just transferring it from one residue onto a different enzyme, replicating the same process. So notice E1 versus E2. So two different enzymes, but essentially the same process. And lastly, step 4 is a ubiquitin ligase. So remember, ligation equals linking. It's just another way of saying joining. So ubiquitin ligase transfers the ubiquitin to a lysine residue on the target protein. So what we have just covered is that the enzymes themselves, the ubiquitin enzymes, first of all bind ubiquitin, but they're not binding the ubiquitin for degradation. They're carrying it in a state that allows it to be linked to the target protein of interest, to the protein that needs to be degraded. So in this diagram, we're going from right to left. So here is our E2 with the ubiquitin attached. Here is our E3 enzyme, and you can see that it is now, again, E2 is closeted by the E3, it's sort of shown encased. Here is our target protein with a lysine residue available, and we now have that ubiquitin being attached. So this group here is attached to the target. So this is our target protein, and we've got one ubiquitin molecule attached. And we could repeat this over and over again, either on this residue or on multiple lysine residues. So again, just to repeat, what have we got here? These steps are just showing you, first of all, look here, this is the activation. How do we actually get the ubiquitin activated? Second step was its transfer onto the enzyme. The third step was its transfer to the second enzyme, enzyme 2. And then the fourth step was the transfer of the ubiquitin onto the target protein. So this is the one that must be degraded. Okay? But remember, we need to repeat this multiple times. Now, we said that this occurs when a protein has reached its end of requirement. And so this is called the half-life. But the question is, well, what determines the half-life of a protein and why are they different? Experiments have shown that it's actually the nature of the amino terminal residue of a protein that is important. So whatever that amino acid is, it dictates the half-life. So at least in yeast, if a protein has methionine at the amino end, it has a half-life of 20 hours, whereas that amino acid is changed to arginine, it's only 20 or 2 minutes. So you can see there's a vast difference depending on the particular amino acid. Why? Because arginine is a destabilizing amino acid and therefore promotes its degradation. So this approach allows the cell to modify the half-life of a particular protein and in turn suit its needs for that particular protein. Now, once the protein has been linked to ubiquitin, it needs to go to the organelle for degradation, and that organelle is the proteasome. So as it says here, proteasome is the organelle that degrades ubiquitin-linked proteins. It has 2 units. It has what is called a 20S catalytic unit and a 19S regulatory subunit. And when you put those two together, they combine to give you a 26S unit. Now, S stands for a Svedberg unit. This is a unit of sedimentation, not mass, not molecular weight or molecular mass. So that's why 20 plus 19 does not give you 39, it gives you 26, because it's looking at the sedimentation of this protein through a viscous liquid. So don't worry too much about that, it's just that you have these different units. So you have a catalytic and you have a regulatory domain. The regulatory domain you can think of as the on-off switch, and the catalytic is where the enzyme activity occurs. So in this case, it's going to be the breaking of the peptide bonds. So as it says here, the regulatory units control the entry of proteins into the catalytic unit. So if they allow it in, it's on. If they prevent it, it's off. And once it's in, you have the degradation involving ATP hydrolysis because it's an energy-dependent process. The ubiquitin is cleaved and is recycled. So remember, ubiquitin equals ubiquitous, which means common. It's present everywhere. So this is a very common protein within the cell, a common marker, because all cells, all proteins are going to have to degrade this or use this for degradation of proteins. So here's the diagram that shows you that idea. Here's our protein. Let's say that its half-life was 20 minutes. So after 20 minutes, 50% has been degraded. That's what it refers to. You can see here we've got 5 ubiquitin molecules on this side, another 5 over here. So there's lots of ubiquitin that has been attached. Protein goes to the proteasome and it's broken down into either peptides or the ubiquitin is released. So we've got the breakdown of that. And then these peptides can then either move on to further stages. And this then goes back to additional proteins. So it's a very simple and straightforward process in terms of the degradation. Now, what happens to those peptide fragments? So as it says down the bottom, potential uses of peptide fragments and amino acids generated from the ubiquitin-mediated degradation. So those peptides can undergo proteolysis, which is breaking of peptide bonds. And this just gives you your individual amino acids, and you can see that here. Now, what happens to these amino acids? Well, the amino acids, they can be left intact if they're required for biosynthesis, which just might be, we want to make that new protein. We degraded that first protein, we want to regenerate that protein. The amino acid can have the amino groups being removed, and the nitrogen is then disposed of by the urea cycle, and this is what we'll talk about later. If this occurs, what you're left with are the carbon skeletons. And the carbon skeletons, as we mentioned above, can either give rise to glucose or glycogen, depending on the needs of the cell. They can go into cellular respiration, where we spoke about pyruvate, or they can generate acetyl-CoA or acetoacetyl-CoA, which is fatty acid synthesis. You can see that there is no wastage that is occurring here. The only thing really is the removal of the nitrogen and the excretion of the nitrogen there. Okay, before we move on, I'll stop here because I want to talk about the removal of the nitrogen. This is where we get into the detail. So let's stop here, and I'll see you in the next recording. Welcome back. So now let's move into the first step. We're at this point here, and we're now going down this pathway. We're going down the amino acid degradation. So we're not using it for biosynthesis. We've got an excess amount, so we need to deal with that amino group, which is linked to the cell central alpha carbon. So nitrogen removal is the first step in amino acid degradation. It is the most toxic component of the amino acid. So the alpha amino group, and remember this alpha amino group is the one connected to the central alpha. So just to draw it again, it is this one here. We are ignoring the contribution here. This is the one that we're interested in. This is the one that, from most amino acids, is going to be transferred to this carrier, alpha-ketoglutarate. And by doing so, it forms glutamate. So the bulk is going to occur here. Now the glutamate that is generated, glutamate is then oxidatively deaminated. So remember, we're going to be losing. Deamination, remember, is loss, just like we had dehydrogenase activity, was removal of protons. Here we're going to lose amine groups to generate the ammonium ion, NH4. And you know that ammonia is a toxic molecule, so is the ammonium ion. And this is where it has to be dealt with by entry into the urea cycle. So this is the initial reaction where we want to deal with excess amino acids. Now, in the center, if I just draw a carbon, say alpha, you can see that this is the amino group, carboxyl group, proton, and R group. So this is the group that I've drawn here. It's exactly the same diagram. This is the group that we're going to lose. So this is transferred, it goes on to alpha-ketoglutarate, and that in turn forms glutamate. The glutamate then loses this group as the ammonium ion, and this is dangerous. But what we have now done is that we have lost that group. We've taken it off the incoming amino acid, we've converted it to another molecule that can be metabolized. So, if we look at the reaction, this is catalyzed by an enzyme called an aminotransferase. So it transfers the amino group. Again, the name is really quite descriptive. These can also be called a transaminase for transfer of an amino group. Same thing, just two different names. And as it says here, these transfer the alpha amino group to an alpha keto acid. So this is alpha ketoglutarate. It's all that they're doing. So it's the same information as we've got here, but we're now showing the movement of this amine group onto alpha ketoglutarate is by an aminotransferase. So if we look at one of the more common ones, aspartate aminotransferase. This is the substrate, aspartate, and it's going to transfer the amino group onto alpha ketoglutarate. So this is one of the most important aminotransferases. So if we look here, aspartate, alpha ketoglutarate, what we have left over becomes oxaloacetate and glutamate. And remember, alpha ketoglutarate goes to glutamate, we saw that above. So therefore the remainder gives rise to the oxaloacetate. And we have seen oxaloacetate multiple times before, such as within the citric acid cycle. Now these reactions are reversible. So they can also be used to synthesize amino acids from these keto acids. We're not going to talk about amino acid synthesis in this lecture or in this course, we're just looking at the degradation. Now we've done that, what happens? So the nitrogen atom is transferred to alpha ketoglutarate, which is what we said, and then it's converted to the free ammonium ion by oxidative deamination of glutamate. And by doing so, we now regenerate alpha ketoglutarate. And this process is occurring through the enzyme glutamate dehydrogenase. And again, a dehydrogenase is leading to removal, and in this case, it leads to the amine group. And this enzyme is interesting in that it can use either NADP plus or NAD plus, which is unusual because most enzymes usually have a preference for one of these coenzymes or cofactors. So this information here, don't want to confuse you. This is just explaining the diagram above. It's telling you the steps and the key players. This is the same process over and over again. Now, in terms of this reaction, it's actually a two-step process. First of all is the formation of a double bond, and then we've got hydrolysis of the Schiff base. And this is really an important reaction, this Schiff base intermediate. So let's have a look. This is some of the chemistry. So remember down here, movement of the nitrogen atom from the incoming amino acid is removed as the ammonium ion. So what are we trying to do? We're trying to remove the amine group of the incoming amino acid, maybe aspartate, and we lose it by generating an ammonium ion right at the end. So here is glutamate. Here is the amino acid, and here is the cofactor, either NAD plus or NADP plus. And because this is a loss, remember we're going to introduce a double bond, we lose protons, and these are captured by these molecules because they're in this particular state, the positive state. And so we see this double bond. So this shift base, or this bond here, this is a carbon with a double bond to the amine group, is called a shift base. And it's called a shift base intermediate because this is part of that original step. We now have this molecule here where we're going to have point of reactivity, and we have a hydrolysis reaction. So the hydrolysis reaction removes this NH2 group, converts it into an ammonium ion, and we now regenerate our alpha ketoglutarate. Remember, alpha ketoglutarate gives rise to our glutamate. So if I go back, this is what we had here. Alpha ketoglutarate was involved, generating our glutamate. So this diagram below is just showing you after that addition process has occurred. We've generated the glutamate, and we're going back to our alpha ketoglutarate. It's just showing you those steps. So again, showing you in another way, we've got our glutamate with the donated amino group, NAD plus or NADP plus, and water. We lose the amine group as the ammonium ion, we gain protons, and we also regenerate our alpha ketoglutarate. So very simple, I think, in terms of the steps and the process in regeneration. Now, what about other amino acids? What we find is that the alpha amino groups of serine and threonine can actually be directly converted to the ammonium ion. Now, we saw that with the previous example, that wasn't the case. We had to go through this process. But in this case, the direct deamination are catalyzed by serine or threonine dehydrotases, respectively. And again, a dehydratase, the DE, is telling you the loss. So it's the loss of that group. Now, this is less critical, okay? But what I want to now talk about is the more critical aspect. If we go back to the aminotransferases, how do they actually do their job? How do they transfer the NH2 group, which is on that central carbon, this one, how do they transfer it onto glutamate? They use this molecule, pyridoxal phosphate, and this is used as the prosthetic group. And again, this is a concept that we have seen time and time again, but remember in the learning objectives, this was the key molecule that we were talking about. Here is pyridoxal phosphate. So you've got the phosphate group here, and I've highlighted here the reactive region. So this is where the transfer of that amine group is going to take place. So if we now look at the mechanism, as we said, aminotransferases contain the prosthetic group pyridoxal phosphate. This is a derivative of vitamin B6. When you go back to your cofactor lecture, you will see this being covered, and we talk about it in the context of vitamin B6. We have an aldehyde group forming a covalent shift base intermediate with the amino acid substrate. And we saw in the previous diagram this shift base intermediate occurring here. This is the important part. The alpha amino group of an amino acid, so remember this is that one we keep talking about, it displaces the epsilon amino group of the active site lysine residue. And I'll explain what that means in a second. But the net result is that the aldehyde now forms this shift base intermediate. So let me just repeat this. The alpha amino group, this is of the amino acid that is to be degraded, just to make sure we're all talking about the same thing. Now we've introduced this new term of the epsilon amino group. What is this? This is part of the R group on the lysine residue. Now, the lysine is within the active site of the aminotransferase. This is the amino acid that is responsible for all the reaction. And so all that we are doing here is that we are taking the amino group and we transfer it onto this other amino acid, lysine. And what you will see is that the aminotransferase likes to be bound to an amine group. So in the off state, it actually binds to its own lysine residue. But because this is the R group position, it doesn't catalyze a reaction. When an amino acid comes in, it stops binding to itself and binds to the incoming amino acid. So it's passing the parcel if you think of it in that way. So that's what this is referring to here as an internal versus an external aldimine. Don't worry about the aldimine terminology. What this is talking about is the internal versus the external bond. So this lysine is part of the aminotransferase. This is part of the actual enzyme. So what is not shown is the whole thing, but you could imagine that this just keeps going and going and going. Then you've got the rest of the protein. So what we have here is that the lysine has an R group, and this R group has now bound to the pyridoxal phosphate, and it's formed this shift base linkage. It's bound to itself. There is no reactivity taking place. When the amino acid comes in, this amino group, this is the alpha amino group, and this one here is the epsilon. Alpha displaces this bond, breaks it, and now is attached to the pyridoxal phosphate. We still have this shift base intermediate bond occurring, and remember, we're then going to lose that amine group as the ammonium ion. So what is the net result? the net result is that our alpha amino acids, remember the alpha amino group, is transferred onto an alpha keto acid. That alpha keto acid was alpha ketoglutarate. And when that occurs, it generates glutamate. We've seen that multiple times. The glutamate then is oxidatively deaminated, so NAD plus, and a hydrolysis reaction leads to NADH and the ammonium ion being generated. And that's really where we've got to. What we're going to talk about in the next recording is now that movement into the urea cycle, so we can now generate urea. So hopefully all of these stages, we've repeated this now multiple times, but we've just added extra layers of detail, will make additional. Okay, welcome back. So, what we've got to now is the ammonium ion being produced. And remember, this is quite a damaging molecule. So it is dealt with by its conversion into urea. So excess ammonium ions are converted into urea by the urea cycle. And it is then excreted in the urine, okay? Now, fish, for example, will also have this process, but they don't need to excrete it in urine because they're in an aqueous environment, it can diffuse out. But they're still using the urea cycle as an example. So if we look at the process, one of the nitrogen atoms of urea is transferred from aspartate. The other nitrogen comes from the free ammonium ion, and the carbon atom comes from bicarbonate. So if we look at the diagram below, these are the steps in the urea cycle. And the cycle begins in this red rectangle, so point 1 as the starting point. And this is where you can see the ammonium ion enters the cycle. So this is a cycle, and also note the regeneration of key components. We will go through this process, but what you can see is that ammonium ion comes in and it binds to carbamyl phosphate. And this is something that we will see time and time again. This is basically bicarbonate with a phosphate group attached. It then combines with ornithine to make citrulline. And remember, this is all now within the mitochondrial matrix. That is exported out into the cytoplasm and it combines with aspartate. These two generate arginosuccinate, which gives rise to fumarate as a byproduct, and arginine. And then through the hydrolysis of arginine, we generate urea. And so remember, those key points we had up here, where we spoke about arginine and other molecules coming into play. So let's go through those steps one by one. First step is coupling, linking, or joining, same term, same meaning, of the ammonium ion to bicarbonate. And this forms carbamyl phosphate. So bicarbonate you have as a buffer within the blood. You've got huge amounts. It is phosphorylated. So the phosphate group is attached. Carboxy, phosphate. The carboxyl group with a phosphate. The amine group displaces this group. Why here and not here? This is more chemically reactive. And remember, that is something that we'll also see in future time and time again, particularly in the nucleotide metabolism lecture. Phosphate groups attached as the point of reactivity. So here it is, carbamic acid. We then repeat the process, and there's an additional phosphate group that is now attached to the other oxygen to give rise to carbamyl phosphate. How does this occur? There's going to be an enzyme. So the reaction is catalyzed by carbamyl phosphate synthetase. So this is the product, and this is the creator. It synthesizes it, hence the synthetase. It is the first committed step in the urea cycle. Therefore, it's going to be the rate limiting step as well. Again, something that we've seen time and time again. The first process is going to control the whole cycle. We saw in the previous diagram that this was in the mitochondrial matrix, and we can see from the above reaction that there were 2 rounds of ATP hydrolysis. So one here and another one here. So 2 ATP molecules make these reactions irreversible. And this would make sense because we're trying to break down the amine group that is no longer required. So you wouldn't want this to go backwards. If we look at the synthetase, carbamyl phosphate synthetase, how is it regulated? It again is regulated in an allosteric manner. So it's active when amino acids are being metabolized for fuel. And we want to undergo a process of degradation. So if there is a lot of biosynthesis, a lot of proteins being made, then this enzyme would be turned off. But if there's an excess and it's being used as fuel, it would be turned on. We now have the carbamyl phosphate, and it is transferred to ornithine to form this intermediate called citrulline. So here we go, ornithine, carbamyl phosphate. You can see the group that is going to be attached. So ornithine carbamylase is the enzyme. Don't worry about the names of the enzyme. I'm not going to be too worried about you knowing that. But you can see that the key point is going to be the transfer of this group here, or the removal of the phosphate, which is shown here. So that's how we know. And that gives rise to citrulline. It's just been added on to this terminal group. And again, you can see that this is an amino acid. Here is our central carbon, amine group, carboxyl group, proton. This is our R group. Citrulline now goes to the cytoplasm, and it does so in exchange for ornithine, and it condenses with aspartate. And remember, condensation is the same as addition. Now, don't forget that aspartate is the donor of the second nitrogen that is found in urea. So this is important. This is why this occurs. So we've got our citrulline, we've got our aspartate that is going to be added, and it's important that there's a plus sign here because this is an addition reaction, condensation reaction. So we've got another synthetase, and you can see that it is just joined back on to this group here. That is the point of reactivity. So you can see that it is a direct addition. And this gives us our arginosuccinate intermediate. This is now quite large, so you can imagine that the next step is going to be cleavage or cutting. And so this gives rise to arginine and fumarate. So you can see that here, arginine and fumarate as the cleaved product. So the fumarate is this region in blue, and the arginine is the remainder. Arginine is then hydrolyzed to generate our urea and our ornithine. That ornithine was part of the process, so it gets transported back to the mitochondrial matrix and the cycle repeats. And again, this recycling and reuse of intermediates is something that we will see often. So again, here's the arginine. We've got arginase, which is the key enzyme that hydrolyzes this process. We've got the ornithine, which is going to be from down here, and we've got the remainder of this group giving rise to our urea. We've got one nitrogen group, we've got another nitrogen group, and then we form the oxygen double bond. And this is now urea. Now, why do we have this? Urea has taken the ammonium ion and converted it into a safer form of nitrogen. We've got two nitrogen groups attached. This allows it to be released and removed effectively. Okay, go through the steps, make sure you understand the process because it's important to understand the transfer and how we're going from something which is quite damaging into something which is much safer. I'll stop here, I'll see you in the next recording. Okay, welcome back. Let's now move on to talk about the next stages. So urea cycle and gluconeogenesis. So an equivalent of 4 molecules of ATP is hydrolyzed to synthesize 1 molecule of urea. So this is quite an energy intensive process. But we also see that we generate fumarate. And this is important because it's a precursor of gluconeogenesis. And ultimately, it can be metabolized to generate ATP. And so this allows us to generate the power for this energy intensive process. It's got to come from somewhere. So remember, none of these intermediates are wasted. By having these byproducts like fumarate, we have the ability to fuel the process and it therefore becomes self-sustaining. That's an important idea, an important concept. So now let's look at that in a little bit of a diagram. We're now looking at the linkage of the urea cycle, citric acid cycle, and transamination of oxaloacetate, and how they are linked by fumarate and aspartate. And we've spoken about both of these intermediates previously. So we started off with the ammonium ion. You join the carbonyl phosphate, you can see the urea cycle here. We're going round and round, and this is the end product. Notice that arginosuccinate, aspartate feeds into that, which gives rise to fumarate, and fumarate then goes to the malate and oxaloacetate, and our alpha keto acid. These can also be involved in the citric, or are involved in the citric acid cycle, which is part of that energy process. And so this is where you have the entry of the alpha amino acid. So by drawing it this way, you can see that it enters in one location, is able to go through. So we've got a bit of a pathway like this, coming from this group through, or we've got the amine group from another location. So very intelligent linkage of key molecules so that they are interacting and are used. So to this point, what have we achieved? To this point, we've basically just taken the amino group away. But then it brings us to the next question. Well, what happens to the carbon skeleton? What happens to the carbon skeletons once the nitrogen atom has been removed? Is it wasted? And of course, the answer is no, it's not going to be wasted. Those carbon skeletons are transformed into metabolic intermediates. And these are going to be important intermediates. And these are converted to glucose, or they're oxidized by the citric acid cycle. So it's another way of fueling either additional glucose or energy through the citric acid cycle. And if you look at this process, there are 7 molecules that are generated from amino acid degradation. And if we look at these will all be molecules that we've seen before, common molecules. So pyruvate, we spoke about alpha-ketoglutarate before, generating glutamate. Succinyl-CoA, fumarate, or oxaloacetate. Now, when these are generated, these amino acids are all called glucogenic because they give rise to these precursors where you can actually generate glucose. So all of these can feed into gluconeogenesis. In contrast, some amino acids that are degraded to acetyl-CoA or acetoacetyl-CoA are called ketogenic because these give rise to ketone bodies or, and it's important to highlight that there, fatty acids. Depending on the location and depending on the requirement, you'll end up with either of these. We'll talk about this in a subsequent, I think, next lecture, but certainly we'll talk about these separately. So you either have glucogenic to make glucose or ketogenic to make ketone bodies. So if we look at this diagram, I'll just zoom out. This shows you the fates of the carbon skeletons of amino acids. So here you can see these amino acids give rise to pyruvate. Pyruvate goes into saloacetate, and that can give rise to glucose. Pyruvate can also go to acetyl-CoA, and that feeds into the cycle. You've got these other amino acids that generate acetyl-CoA or acetoacetyl-CoA, which is essentially a breakdown product. You've got two acetyl-CoA units joined together to form this one unit. And these come together to form your ketone bodies or fatty acid synthesis molecules. So you can see, none of this is wasted. You don't want to waste those carbon skeletons in this process. Okay, near the end of the lecture, I'll stop here because I want to talk about inborn errors of metabolism. And in this case, we're going to talk about amino acid metabolism defects. Okay, so in the last part, I want to talk about those inborn defects associated with amino acid metabolism. And here we're talking about phenylketonuria or PKU. And so this is an example of a disease affecting amino acid metabolism. And it's caused by a deficiency of an enzyme called phenylalanine hydroxylase. Now, because we know that these enzymes can have cofactors, this can also be due to the absence of the cofactor, which is tetrahydrobiopterin, although this is less common. So just think, it's the enzyme more so, but it can also be the cofactor. Because when an enzyme requires a cofactor, if one is missing, the net effect is going to be the same. Now, without this enzyme, what happens? Well, as you take phenylalanine into the body, it's going to build up in all of the bodily fluids because it's not being metabolized. And ultimately, it gets converted into metabolites such as phenylpyruvate. Now, what I want to show you are the differences in metabolism that can occur. The first one we have covered at length. This is the normal process. So this would be the rate limiting step because this is the first process. A goes to B. So substrate gives rise to a product. That product is now the substrate of the next reaction, so on and so on, and it's nice and linear. When you have an absence of an enzyme, you have feeding in of the pathway. This enzyme might be the hydroxylase. It is not present, so therefore this builds up, and the concentration goes higher and higher. As the concentration goes higher and higher, it needs to be dealt with by the cell, and it goes into a different product, it goes into a different pathway. But this gives rise to abnormal metabolic products. These are unexpected reactions. And as a result, they can give rise to toxic intermediates. That toxic intermediate is what leads to disease. And this is what we're talking about. We're saying that the hydroxylase is missing, so therefore, rather than going through for the normal metabolism, it's metabolized to phenylpyruvate, that goes into a different pathway, and that is what causes disease. So here we can see phenylalanine. Here is the central alpha, so this is the amino group to there. It goes across to the alpha keto acid, and you have the alpha amino acid being released, and you end up with phenylpyruvate. And this is now the problem. Now, in terms of this disease, if it is untreated, PKU is actually quite serious. It will cause mental retardation and actually early death in the affected children. It is treatable where you have a diet that is very low in phenylalanine, but that can be very expensive because you have to eliminate all of that amino acid. It is a genetic disease, so it is an autosomal recessive inherited disease. It's actually fairly common, 1 in 10,000 newborns. And we'll see why that's important in a second. If the individual is heterozygote, which means that they have one normal and one affected gene, they appear normal because they have the normal hydroxylase enzyme. If they have both that are affected, then they're going to present with the disease. As a result of this, so something that is inherited, is very common, and can be treated, all newborns are tested. So it is a serious condition, it is common, and it is treated. These are the criteria for the testing for this particular disease. If it was not serious, it would not warrant testing. If it was extremely rare, it would warrant testing. And similarly, if it couldn't be treated, well, then there's really not a lot of point of its testing or identification. So this is something that is used very commonly. Okay, going to stop there. A lot of content, but I think it's all quite logical and it makes sense. So here is your summary section, and of course you've got your tutorial questions. Hi everyone, and welcome to lecture 16. So as you see here, this lecture we're going to be talking about lipids, and lipids are the correct terminology that we would use for fats. So we're going to talk about why fats are important within the body and how they're used. So if we look at the study aims, you can see that we'll talk about the biological roles of lipids. We'll also talk about the nomenclature, so how we talk about fatty acids and how they're referred. And we'll also then move on to talk about how fatty acids are released from triacyl glycerol. This is really the storage form of fatty acids within the cell. We'll then move on to talk about the 6 stages required for beta oxidation, and this is a specific form of oxidation that relates to fatty acids. And excuse me, you'll get huge amounts of ATP generated from fatty acid oxidation because they can undergo many more rounds of reduction. And as a result, you have to be able to calculate the amount of ATP molecules that are generated through beta oxidation. But this is actually a really easy calculation to do, so don't worry about that. We'll then finish off by talking about the importance of ketone bodies, and this will really link into some of the work that we've done previously with the citric acid cycle. And right at the end, we'll talk about how fatty acids can be used to generate other biologically active molecules, and these are very, very potent and control numerous functions within the body. So it gives you another layer of appreciation of what fatty acids can be used apart from a store of energy. So space for the questions, and there you can see the reference to your lecture notes or your textbook. So as we start off, I always like to give you an introduction. So remember, we've talked about lipids and fats. These two terms can be used interchangeably. Lipids is the scientific term, fats would be the lay term, but we know that these have got lots of important roles within the body. And so if we think about them, what would these be? The first ones are building blocks for the cell membrane. The cell membrane is made up of lipids, and more specifically, these are referred to as phospholipids or glycolipids. So phospho is telling you that they have a phosphate group, glycolipids are telling you that we've got carbohydrates attached. And we have seen the addition of these groups onto other molecules throughout this semester, so this should not come as a surprise to you. We also can see that some proteins require fatty acids to be covalently attached. And by doing so, this actually targets them to the cell membrane. So remember, like will dissolve like. So if you put a fatty acid onto the protein, the logic is that that fatty acid will help locate or target that protein to the cell membrane because that's where its activity is required. For example, if it's part of the signal transduction cascade. We'll also look at fatty acids as derivatives of hormones, so they can be synthesized and degraded into various hormones. These are collectively referred to as eicosanoids, and this refers to a specific fatty acid precursor, but this is the broad term that we'll talk about. And the one that most people will already know is that fatty acids are the main energy reserve within the cell, within the body, and the term that we use is a triacyl glycerol. So there are 3 acyl groups that are linked to a glycerol backbone. We'll see that in the diagram below. These can also be referred to just generally as fats, as we've seen, but also as triglycerides. So again, just referring to those 3 units that are attached. Now, if we look here, we have our glycerol. This is referred to as the backbone. And we have the first position. This is called the SN1 position. The second position is SN2. And then the third position is an SN3. You can have 3 fatty acids linked there. That's where you would have glycerol as a triglyceride. But what we also have here, this is now showing you in this diagram a phospholipid. So we have our phosphate group attached to an alcohol group, and this is the phospholipid group. So you can see here basic structure of a phospholipid, backbone with 2 fatty acids attached, and the head group, which is the phosphate or the alcohol group. So if we want to go into a bit more detail in terms of the different common head groups, so as you can see down the bottom here, common head groups that are found in phospholipids, we've got glycerol as the backbone, so this is shown in blue, so we can see the 1, 2, 3 carbons that make up the glycerol. And we've seen glycerol in the previous lecture where we were saying this is a non-carbohydrate precursor that can be used in metabolism. In green, we can see the first 2 fatty acids, so R1, R2 just refers to the different types of fatty acids that will be attached. And then here we've got the phosphate group shown in black, and then the alcohol group shown in red. And you'll notice that there are differences along the 4 that are shown. So we've got phosphatidyl serine, we've got phosphatidyl choline, phosphatidyl ethanolamine, and phosphatidyl inositol. These last terms refer to these groups here, okay, as the different groupings that are around. This one in particular, phosphatidyl inositol, you'll see this in the signal transduction lecture, because this inositol group can be released, and this also acts as a signaling molecule. Can I also just highlight for you down here, these are shown for information only. You don't need to memorize them. You need to be familiar with their terminology and what they are, but certainly you don't need to know the structures of these groups. That's not important at this level. Okay, so if we move on to talk about the nomenclature and the numbering, because fatty acids, just like any other chemical chemistry form, there's a way of referring to these. There is a systematic name that is used, and this is derived from the parent hydrocarbon. So fatty acids are essentially just carbon and hydrogen molecules. So they're basically hydrocarbons. And what we see is that we substitute the last term of OIC for the final E. So we can see here that we've got an 18 carbon fatty acid with 0 double bonds. That's what this is referring to. This is octa-decanoic acid. So 8 and 10 makes 18, decanoic acid. And this is referred to as saturated, which means that there are no double bonds. So what that means is that all carbon atoms have a single carbon-carbon bond, or carbon-hydrogen bond. Okay? Here we've got 18-1, so we've got 1 double bond. This is octa-decanoic acid. 18-2 is octa-dianoic acid with 2 double bonds, and so on. So it's very descriptive. It's very easy from a chemistry perspective. We also have directionality. So fatty acids are numbered from a carboxyl length. There's a carboxyl length, and this is the starting position, so you go 1, 2, 3, etc., until we get to the other end of the molecule. We also have double bonds, and these double bonds can exist in different locations. So the position of the double bonds is represented by the delta symbol. So this tells you the location. And then you've got the number to be more specific. So thank me, so I'll just write that one in. So that would then tell you delta. So this is your CH3, as you can see here, just the opposite configuration, and this is shown as the omega end. You can see that the first CH2 group is referred to as alpha, the next one is beta, and then the length of this will change, and the terminal is going to be omega, which is down this end. just again highlight to you the beta carbon. This is important because this is where oxidation is going to take place. It's going to be onto the third carbon, so the second carbon from the carboxyl group. So by numerically, this will be the third carbon where we start to cleave that particular fatty acid. And we'll see this over and over again. The alternate way of referring to this actually refers to the position of the first double bond. And we actually work this out from counting from the opposite or the omega end, okay? So it's the other direction with the methyl carbon. And so in this case, that's now referred to as the 1. So omega-3 fatty acids, and this is something that we will see often. So omega-3, that way, you can write it the same way. Have their first double bond on the third carbon atom. So typically, in the previous example, we started from here, we said 1, 2, 3. Remember that this was alpha, this was the beta carbon. If we're using this nomenclature, we're actually starting from this carbon here. So we're saying, okay, the first carbon, second carbon, third carbon, this is where the first double bond is. So therefore, this is an omega-3 fatty acid. This is probably something that you'll have heard about. You'll have all heard about fish oils. Fish oils being high in two fatty acids called EPA and DHA, and these are both omega-3 fatty acids. So this is what it's referring to. It's just saying from the omega end, where is that first double bond? And then based on the valence, you can then work out what the subsequent bonds are going to be. Okay, let's just finish off some of the characteristics of these. So there are variations in fatty acid chain length and also the degree of unsaturation, and this just means the number of double bonds. So it will vary. So fatty acids usually have an even number of carbon atoms. Typically it's between 14 to 24. You can have smaller, you can have larger, but this is the average size that we will see. The chain is normally unbranched, so therefore it is a linear chain. So that is something that is quite different, say, compared to glycogen, where we had branching on purpose, and that was part of the activity. Fatty acids are linear. We've already seen that fatty acids can be saturated, so there's no double bonds, or they can be unsaturated, having various numbers of double bonds. So we've got various numbers of double bonds, and obviously they can actually occur at different locations, as we saw with the omega, for example. Double bonds are normally in the cis configuration, and the chemical properties of a fatty acid depends on two characteristics, and these are the chain length, so more carbon atoms, and also the degree of saturation, so the number of double bonds. These will affect their activities. So if we look at that, we can see that unsaturated fatty acids, so fatty acids with a double bond, have lower melting points than saturated fatty acids of the same length. So what does that mean? That means when you've got an oil, these are typically unsaturated fatty acids. When you've got solid fats, like butter, these are more likely to be, saturated fats. Okay, so these are the two basic differences. Now, because you can have a mixture of unsaturated and saturated fatty acids, this actually affects the cell. So short chain length and unsaturation will actually make the cell membrane more fluid, and therefore you'll also vary the derivatives that come from that. So the cell is able to change the ratio of unsaturated and saturated fats to either make the membrane, if it is more unsaturated, it's going to make it more fluid. If it is more saturated, then it's going to be more rigid. And we actually know that depending on the fluidity state of the membrane, that actually can affect the health of the cell, in turn the health of the organ, and therefore the health of the individual. So this is quite an important structure that we need to cover. Okay, I want to move on now to talk about triacylglycerol, but I'll do that in the next recording because we're going to start to talk about oxidation and its energy. So I want to do that in a separate recording. So see. Okay, welcome back. So as we just finished, we finished off by talking about triacylglycerol, or TAGs. So remember, tri is 3, acyl is a way of referring to a fatty acid, and the glycerol is the backbone that we saw in the very first diagram. So triacylglycerol is used as a concentrated store of metabolic energy. This is how we store fuel for energy purposes. We know that we can't do this for glycogen because we have limited reserves within muscle. This is the way that we store long-term and greater amounts of energy. Why do we get so much energy? We get so much energy because it is highly reduced. So this means it has many rounds of oxidation. It's able to undergo many rounds of oxidation, which means that you get more rounds of ATP being produced, and it is also anhydrous, which means that it is lacking water, which again just facilitates that oxidation. We know that triacylglycerol is stored in adipose tissue. This is the storage around the body. And when we look at the difference between, say, fatty acids and glycogen, we can see that the amount of energy is not even comparable. Fatty acids are more highly reduced, so we've got a gram of fat. It will give you 6 times as much energy as 1 gram of hydrated glycogen. So remember, hydrated makes it less energy efficient, and we know that glycogen is not as reduced, so therefore it doesn't go through as many rounds. So if you wanted to have glycogen as your energy store, you would need to have 6 times as much just to have the equivalent amount of fat. And you can see that obviously that's going to make it untenable. So if someone had 10 kilos of fat on their body, they would actually need to have 60 kilos of glycogen, and that's obviously not going to be very useful from a perspective of the individual. So we know that we've got fatty acids there. So how do we actually access those fatty acids in triacylglycerols? They're hydrolyzed, they're released through a process of hydrolysis, and the enzyme that does this is called a lipase. So we know ASE is the enzyme, so this is the enzyme that just cuts the fatty acids away from the triacylglycerol. So the initial event in using fats as energy has to be the release of the fatty acids through hydrolysis by the action of the lipase, because otherwise the fatty acid is still linked in the triacylglycerol. It can't undergo oxidation, so we need to cleave it, and that process is referred to as lipolysis. It's just cutting those individual fatty acids away from the triacylglycerol. And the overall net reaction is very simple. You've got one triacylglycerol, you've got 3 fatty acids that are attached, so therefore you need 3 water molecules. This gives rise to the free glycerol backbone, your 3 fatty acids and 3 protons. And this can then undergo beta oxidation. We know that glycerol is a non-carbohydrate precursor, so this can be used to make glucose, as an example, in gluconeogenesis. So none of this is going to be wasted. Now, in terms of the lipases, how are they controlled? They're going to be controlled by hormones. And so we've got the 2 hormones that we've spoken about previously. We've got adrenaline and we've got glucagon. Both adrenaline and glucagon will promote the breakdown of fat. How do they do this? They have to transmit signals. So for these to work, they need to involve a signal transduction pathway. They need to be able to communicate to the cell. In this case, these 2 hormones will activate an enzyme which is called adenylate cyclase. And we've touched on this previously in an earlier lecture. And you should remember, adenylate cyclase increases the intracellular level of cyclic AMP. That's what this is, cyclic AMP. So this is adenosine monophosphate. That terminal phosphate makes a circular bond. Cells are very sensitive to the level of cyclic AMP. So as cyclic AMP goes up, it stimulates the activity of a protein kinase called protein kinase A, and the A refers to the AMP. This in turn activates the lipases. The lipase then hydrolyzes the fatty acid, and so releases it. Now, if you have a forward reaction, you need to also have the controlling reverse reaction. So the reverse reaction is going to be insulin. So insulin will inhibit lipolysis, and therefore it promotes fat storage. So anything that is going to increase the level of insulin, such as glucose, is going to promote fat storage. Now, once those fatty acids are released, how are they transported? How are they brought around the body? Now, you know that fat and water are not miscible, and we know that we are mostly water. I think 70% is always the number that's spoken about. So how do we transport fatty acids in an aqueous environment? We do so by linking it to a protein within the body called albumin, and this is the major protein that is present in the blood. And this is one of its major functions, is to transport fatty acids throughout the body in a state that is compatible with an aqueous environment, so it doesn't clog up the blood vessels. Now, we've got lots of different tissues that will use fatty acids to make ATP. As I mentioned to you before, glycerol can be used to make glucose through gluconeogenesis. This is going to occur within the liver, and the liver will also break down fatty acids to make ATP. Now, this fatty acid albumin complex, though, cannot cross the blood-brain barrier. Where it's missing, so the brain is dependent on glucose. And again, we have spoken about the importance of glucose in the brain time and time again. So let me just zoom out, because this is showing you the hydrolysis of fatty acid molecule. Okay, that fits in. So here is our triacylglyceride. Remember, this is not a phospholipid, we're just talking about the triacylglyceride. So here is carbon number 1, carbon number 2, carbon number 3, and we've got one fatty acid, got a second fatty acid, and the third fatty acid. The nature of the fatty acid is shown as R1, R2, and R3 because it actually doesn't matter. We'll talk about specific fatty acids, but in this diagram it actually doesn't matter. So we have the lipase, and if it occurs 3 times, you'll see that it is going to cleave the fatty acid there, I should say, not that spot there. Okay? And so what this will give you is your glycerol molecule and your 1, 2, 3 fatty acids. And those fatty acids can then go through beta oxidation, which we'll talk about later in the lecture, and give you huge amounts of ATP. So that's the lipolysis process. So let's talk about degradation then, start to move into this pathway. So we've seen that lipolysis generates glycerol and fatty acids. And we know that both of these products can be metabolized for energy, and we'll see that in the diagram below. And we know that the liver will take the glycerol, and either through glycolysis or gluconeogenesis, it will be acted on. Other tissues, though, will metabolize the fatty acids through beta oxidation, and this is going to give rise to lots and lots of ATP, and it's how the cell is able to move forward and meet its energy demands. So here is the diagram that summarizes all of these potential fates of glycerol and fatty acids. So remember, we've covered most of this, but remember, TCA, or the citric acid cycle, tricarboxylic acid cycle, is all the same, and we've spoken that at length in its own lecture. But if we've got a fat cell, it's storing the triacylglycerol. If we've got adrenaline that is being produced, the triacylglycerol is cleaved, and we will release glycerol molecule and fatty acids. The glycerol goes into the circulation, will go to the liver, and through glycolysis, it can be used to be converted into pyruvate, or through gluconeogenesis, it can be converted into glucose. The fate or the direction of glycolysis or gluconeogenesis we've covered before, but remember, if you have a lot of glucose and you want energy, then you go down the pyruvate pathway. If you don't have enough glucose, then you want to synthesize it and go through the gluconeogenesis pathway, but there is no wastage. The fatty acids that are released, and so we've got a list, well, maximum of 3, these will go to other tissues, and they will undergo fatty acid oxidation. When they undergo oxidation, you generate a lot of acetyl-CoA. Acetyl-CoA feeds into the citric acid cycle, and we know that gives rise to CO2 and water, and then through the electron transport chain, we are going to generate a lot of ATP. So there's a lot of precursors generated here that feed into the electron transport chain, which then means that we make lots and lots of ATP, as we will calculate later. Okay, let me stop there because I'm going to go into the detail of fatty acid oxidation, and we'll do that in its own separate recording. So see you in the next lecture. Okay, welcome back. So now we're going to talk about fatty acid and its oxidation. Now, in order for a fatty acid to be oxidized, it must first be activated. And what this refers to is the addition of a phosphate group. So whenever you see the word activation, typically it means the addition of this phosphate. So activation involves ATP. ATP is going to give us that phosphate group because that's where it's more likely going to come from. So fatty acids react with coenzyme A to form acyl-CoA. And this reaction occurs on the outer mitochondrial membrane. So not surprisingly, we're in the mitochondria for this process to occur. So if we look at the diagram below, this is showing the activation of fatty acids for metabolism. These are the steps that are required. So we've got a released fatty acid. That's what this is showing you. So glycerol has been generated, and we've got at least another 2, plus this one, of fatty acids that have been released. They interact with ATP, we form this acyl adenylate. So we have this AMP group. So rather than being phosphorylated, the action of ATP is occurring here. So we've still got that group, so we've still got this terminal phosphate there. This makes this now the most reactive component, and it then interacts with the sulfurhydryl group of CoA to make an acyl-CoA. So remember, the whole point of adding the AMP, or involving ATP, was just to direct this group and its addition onto that carbon. And so we have the sulfurhydryl group linked to the CoA and the regeneration of AMP. So this is important. So remember, we've got the displacement because we've got the targeting of that location. Once that occurs, we then have transportation. So the activated fatty acid is transported across the inner mitochondrial membrane. And the only way that it can do this is it requires the fatty acid to be linked to another protein, which is called carnitine. And carnitine is a derivative of glycine, which is just a single amino acid. So again, we can now start to see the interaction that's occurring with amino acids, not just being proteins, but also other intermediates that are involved in more complicated types of metabolism. So carnitine is only a carrier, so it is exchanged, it's not consumed. It is exchanged for coenzyme A to form acyl-carnitine. The acyl-carnitine is then transported across the membrane. The carnitine is regenerated, and what we've now achieved is that the acyl-CoA, the fatty acid that's been activated, is now in the inner mitochondrial matrix. We've got the regeneration of the carnitine, but we've been able to bring the fatty acid internal to the mitochondrial matrix, where it can undergo beta oxidation. So when we come to talk about oxidation, do not forget these steps. And importantly, do not forget that we have used or consumed one ATP molecule at this stage. So when we work out the amount of ATP, we need to account for this ATP here. Now, here is the process. So this is how we transfer the activated fatty acid across the carnitine, and this, remember, allows the movement of the fatty acid into the inner mitochondrial space. So here's our acyl-CoA. Remember, this is the fatty acid, this is the CoA group, here is our carnitine, and we displace, we have the bonding occurring, so we've got the displacement occurring, so we end up with our acyl-carnitine, and the group is now released. So the fatty acid is now up here. I'll just show the oxygen because that's where it was with the sulfur group, and here is the carnitine molecule. Now, from a diagrammatic perspective, this diagram is really quite nice. It shows you the carnitine, the acyl-CoA, that displacement, and the formation of the acyl-carnitine, which is what the diagram above is showing. So here's the acyl-carnitine, it's the same thing. This is the location in the diagram where we are. This is the cross-section, so this is the cytosolic versus the matrix side of the mitochondrion. And so we've got the transfer across. Once it's on the inner or the matrix side, the acyl-carnitine, the CoA is put back on, and the carnitine is regenerated, and it is able to freely translocate across, and we use a translocase. So this has just been recycled going round and round. But the net result is that the fatty acid that was on the outside is now on the inner mitochondrial matrix side, and this is where beta oxidation is going to take place. So let's go into the steps of beta oxidation. So the first 2 steps that we just saw are still part of beta oxidation because they're required to activate the fatty acid and actually bring it into the right compartment within the mitochondria. Now, beta oxidation is the degradation of fatty acids. So that's what beta oxidation is referring to. And we're going to be cutting 2 carbon atoms every single cycle. So every cycle, we chop off 2 carbon atoms. And by doing so, we generate acetyl-CoA, and this then enters the citric acid cycle, or the TCA cycle. Remember, all the same thing. The beta carbon is the second carbon from the terminal carboxyl. We saw that in the previous diagram, but here is another example. Here is our carboxyl group. We said this is carbon 1, carbon 2, carbon 3. And the other notation is this is carboxyl, so this is alpha, this is beta. So this is the carbon where beta oxidation is going to take place. Now, there are 4 steps after the fatty acid is entered or enters the mitochondria. First one is activation of fatty acid. So again, we're seeing activation, so there's going to be use of energy. So fatty acid is oxidized to introduce a double bond, and here we've got FAD being used. The double bond is now the point of reactivity. It becomes hydrated to induce an oxygen. The alcohol is then oxidized to a ketone, and we've now got NAD+. So we've got two different cofactors being used, or coenzymes. And then what we end up with is a 4-carbon fragment, which is then cut or cleaved by coenzyme A. And when this occurs, we generate acetyl-CoA, and the net result is that the fatty acid is now 2 carbon atoms shorter. This equals 1 cycle. That's 1 cycle of the entire process. So let's go through those reactions. So the first reaction we said was oxidation of an acyl-CoA, so a fatty acid, by an acyl-CoA dehydrogenase. And we know that this is going to strip off protons. And so what this gives us is an enol-CoA with a trans double bond at 2 and 3, because remember, these are the points of reactivity, the third carbon of the fatty acid. FAD is reduced to FADH2, so there are 2 protons that are removed. So look at this diagram. You can see here we are. We've got 1, 2, and 3. So this is 1, 2, and 3. Acyl-CoA, we've got the oxidation, so the FAD gains the protons. We strip a proton from here and a proton from here. To maintain the valency, we now have to introduce this double bond. And this is what we see here. Now, when we look at this, this is going to be a point of reactivity. Could also be here, but because this is going to be an enzyme-driven process, we're going to have the reactivity of this carbon double bond as opposed to elsewhere. Second reaction, that double bond that was just introduced, so C2 and C3, so the one we were talking about, is hydrated by enol-CoA hydratase. So it's going to add water. So this is what we had on the previous diagram. We had our water, and it is reacted at that particular location. We've added a hydroxyl group. And again, the valency is maintained. Third step was another round of oxidation. So this is where we convert the hydroxyl group into a ketone group. And by doing so, we generate a molecule of NADH. So again, this is the group that we had above. We've got NAD plus going to NADH, and we lose a proton through oxidation. And remember, this hydroxyl group gets converted to this ketone. And we maintain the valency here, so this carbon has still got 4 bonds. And then the last one is going to be the thiolysis or the cleavage step. So this is reaction number 4. So we've got cleavage by a thiol group from a second molecule of the CoA. So remember, sulfur-hydryl group is also referred to as the thiol group, which is the coenzyme A. And what this gives rise to is acetyl-CoA, which we said goes into the TCA, or the citric acid cycle. You've got a fatty acid that is now 2 carbon atoms shorter, and that fatty acid can then undergo another round of beta oxidation. It just keeps going on and on and on until the end of the oxidation process. So as we see here, the ketoacyl, as we had before, and we had this group being added, the acyl-CoA comes in, it lyses the molecule, and we break it apart. So we end up with our acetyl-CoA. So this group here is converted to CH3, and this group is formed. This group comes down here. This is our acetyl-CoA, and our acyl-CoA has been shortened by 2 carbon atoms. That is 1 cycle of beta oxidation. This is the summary of all of those steps. Now remember, these 2 steps were before oxidation. These were involved in actually activating the fatty acid and bringing it into the mitochondria. These further 4 steps were actually the oxidation, and where we got the beta oxidation taking place and the energy being generated. So this is the important stages. So they're all important because without the first 2, none of these subsequent stages would occur. Okay, I'll stop there because the next part is actually going to be a bit of a mathematical calculation. It's very simple, but we want to work out how much energy is generated from Okay, welcome back. So we just went through the steps of oxidation. And the next question is, well, okay, you said to me that there's lots of rounds of oxidation, I can see that, but how much energy is actually made by the complete oxidation of a fatty acid? Now, I could pick any saturated fatty acid, but in this example, I've picked 16,0. And you'll see, once you understand the maths, it doesn't matter what this number is. It's very, very easy to calculate. But there are 1 or 2 points that you need to remember, otherwise you'll make a mistake. So in each round of beta oxidation, what do we generate? We end up with an activated fatty acid. So remember, acyl-CoA is just the activated fatty acid, what we're trying to metabolize. It becomes 2 carbon atoms shorter. We make 1 molecule of FADH2, 1 molecule of NADH, and a molecule of acetyl-CoA every cycle. So what we then have to work out is, okay, well, for a given fatty acid, how do we work out the number of cycles? And it's actually very simple. It's just the number of carbon atoms divided by 2, and then we subtract 1 from that number. So if we said 16 divided by 2 would be 8 cycles, we subtract 1, it's 7 cycles. Now, this is where students will get confused, which is why I put this in bold. Why is it 7 and not 8 cycles? Think about the very last cycle. The last cycle is a 4 carbon molecule. If you have the thiolysis step, it cuts it here. We end up with acetyl-CoA. What's going to be on the other side? It's going to be another acetyl-CoA. This is already a 2 carbon molecule. It does not need to go through another round because the end result is just going to be this molecule. So the very last cycle gives you 2 extra acetyl-CoA. So that's why we subtract 1 actual cycle, because the last one doubles the amount that is produced. So if I had another example, 20 carbons. 20 divided by 2 would be 10, minus 1, we would say that there are 9 cycles. Okay, it's very, very simple. Now, this is important for most of the steps, but also this number we must not forget, because remember, we get extra product at the end, and this is important to account in our calculation. So if we go through the steps, we know the value of the NADH and the FADH2 that are generated, because we've seen this in the electron transport chain. FADH2 gives rise to 1.5 ATP molecules. NADH2 is slightly more, so 2.5 ATP molecules. So how much are we generating? Well, we said the number of cycles was 7. We times 7 by the 1.5 ATP molecules, because we know that there's going to be 7 molecules of FADH produced. 7 times 1.5 is 10.5 ATP. We know that there are 7 cycles for NADH being generated, so 7 times 2.5 is 17.5 ATP molecules. Now, what about the acetyl-CoA? Acetyl-CoA is valued at 10 ATP molecules. Now, remember, at the very last cycle, we said that we ended up with 2 extra acetyl-CoA. So this is where we now say that it's actually equivalent to 8 cycles. It hasn't actually had to go through 8 cycles, but we have to account for that extra molecule. So we would say 8 times 10 is 80 ATP. And we then add those up. So 80 plus 17.5 plus 10.5 is 108 ATP. But don't forget, we used 2 phosphate molecules at the beginning, and we used them because we went from ATP to AMP. And we released pyrophosphate. You've got 1 phosphate, 2 phosphate molecules. These 2 phosphate molecules have been used. These were the ones that I said were consumed right at the beginning. Go back to the lecture recording where I highlighted those. Now, you have spent 2 ATP, so the net result is not 108, but it's actually going to be 106. So 2 minus 108 is 106 ATP molecules final. That is important. So make sure you go through the process of this calculation. Make sure you understand the number of cycles, the logic as to why the last cycle gives you 2 of the additional acetyl-CoA molecules, and then how they're accounted for in this last stage. Once you do this, the maths is super simple. And the only other thing is not to forget the consumption of ATP at this particular point. That's a really common mistake that students will make. They'll either get the number of cycles incorrect, or they will forget that they have used ATP in one of the very first stages. Okay, I'll stop there because I wanted to just focus on this calculation. Make sure you understand it. Make sure you highlight this in the lecture or tutorial if you don't understand it. Okay, welcome back. Let's keep moving on with the metabolism of fatty acids, and now we're going to have to move into talk about ketone bodies. These are another source of fuel that are derived from fats. And so what we know is that acetyl-CoA produced by beta oxidation enters the citric acid cycle. That's fine. Acetyl-CoA can be used to form alternate fuels, and these are called ketone bodies. And these are referred to more specifically as acetoacetate, D3-hydroxybutyrate, and acetone. These are all collectively ketone bodies. Now, these can be used as fuel, but they generate less ATP, they're water-soluble, and therefore they're easy to transport. So even though they're less ATP, this water solubility and ease of transport is of benefit. And ketone bodies are important fuels for the heart and skeletal muscle, and actually in preference to glucose. So these are important molecules that are used by the particular tissue. Now, where are these made? Not surprisingly, these are going to be in the mitochondria, but the mitochondria of the liver. And there are 3 reactions that generate these ketone bodies. The first one is that we get 2 molecules of acetyl-CoA combining or condensing to form acetoacetyl-CoA. So this is just acetyl-CoA plus acetyl-CoA. And when you get that, it's referred to as acetoacetyl-CoA, because you've got the aceto group here and you've got the acetyl group there. It's just that addition. So remember, combining, condensing is the same as addition. That condensation of acetoacetyl-CoA occurs again. So we have another acetyl molecule, acetyl-CoA, coming together, and we form this lovely molecule here, beta-hydroxy, beta-methylglycerol-CoA. Don't worry about the name, just remember this abbreviation, HMG-CoA. This is an important intermediate in this process. Once this is made, HMG-CoA, it is degraded to acetoacetate. And remember, we said up here, acetoacetate was a ketone body, and we release acetyl-CoA. The acetyl-CoA could then be recycled to make another ketone body. Acetoacetate is decarboxylated, forms acetone, which is the other one that we had up here, and we release CO2, because remember, this is a carboxylation or decarboxylation, so we're going to be losing the CO2 molecule. Now, in individuals that are undergoing a lot of ketone body production, this can lead to a process called ketosis. And this is where we've got acetoacetate being produced faster than it's being used. And we see this in diabetics. So remember that the metabolism in diabetics is uncontrolled, that you've got glucose being produced even though there is excess glucose within the blood. And as a result, we have a lot more acetone being produced. And you can actually, this is a diagnostic marker, you can actually smell the acetone on a diabetic person's breath that is undergoing ketosis when they're using a lot of ketone bodies as fuel. So let's go through those processes. So remember, the first one was 2 molecules of acetyl-CoA condensing to form acetoacetyl-CoA. So here's the first molecule, here's the second. They come together, they are condensing, and they join together. We then had another molecule of acetyl-CoA condensing to form the HMG-CoA. HMG-CoA. And here we have it here. So another addition of that particular group. And then we had the cleavage. So the HMG-CoA loses an acetyl-CoA to form acetoacetate. And that is this molecule here. This is the acetyl-CoA group there. And that obviously can go back to the beginning to start the process again. Now we've got this sort of fork in the road approach. Acetoacetate is decarboxylated to generate the hydroxybutyrate and the acetone. So depending on the location, so here's the decarboxylation, generates acetone. Here we've got the oxidation, because we're losing protons to form hydroxybutyrate. I've got these shown in different locations because they will give you different products. I will talk about those in a moment. So how are these ketone bodies used as fuel? How does this work? As we just saw, ketone bodies lead to the generation of NADH and ultimately acetyl-CoA. That acetyl-CoA feeds into the citric acid cycle, and that ultimately gives rise to our ATP. So it's really these precursors and their linkage to the citric acid cycle. So if we look here, this is the basic process. We had our acetoacetate. This is converted through a transferase to succinate, which we have seen in the citric acid cycle. It's involved. That gives us acetoacetyl-CoA, a thiolase. Remember, acetoacetyl-CoA was just 2 acetyl-CoA units. So thiolysis will break them. And we end up with our 2 acetyl-CoA units that go into the citric acid cycle, tricarboxylic acid cycle, which ultimately, through the electron transport chain, is going to give rise to lots of ATP for that cell. Okay, I'm going to stop there because we want to now finish off by talking about some of the other non-metabolic or non-energy driven metabolic processes for fatty acids. So see you in the last recording. Okay, last recording. Do fatty acids have other biologically important roles? And the answer is absolutely. So please don't go away thinking, well, fatty acids is just used as part of the cell membrane or as a store of energy. They can also be used to make other biologically active precursors. Now, one of the most common fatty acids in a triacyl glycerol is called arachidonic acid. And it's a 20 with 4 omega-6 fatty acid. So there are 20 carbons, 4 double bonds, and the first double bond is on the 6th carbon. This is very common in animal fats, whereas the omega-3s you see mostly in fish oils, or fish oils are rich in omega-3s. They're present in other foods, but this is where we see really the discrepancy or the difference between the two. This fatty acid is released from triacyl glycerol through activation, but this is not activation for energy, this is activation for making other biological molecules. And so the enzyme that does this is a lipase, but it's called a phospholipase, and more specifically phospholipase C. There's A, B, C, D, etc. We're just looking at this one because it cleaves at a specific location. The free arachidonic acid that is generated can be metabolized to generate eicosanoids, and this, remember, is just a family of products. So it's a very broad term for all of these products. But what does it mean? Icosa, the prefix, refers to molecules with 20 carbon atoms. And as we saw up here, arachidonic acid is a 20 carbon fatty acid. This arachidonic acid is metabolized through different enzymes, through the cyclooxygenase enzymes to give rise to prostaglandins and thromboxanes, and also the lipoxygenase enzyme to give leukotrienes. These groups within here, there are multiple different derivative variants of prostaglandins, thromboxanes, and leukotrienes. But all of them are very, very active, and they mediate lots of different processes, such as vasodilation, vasoconstriction. And you can see that these are two opposing things, aren't they? So dilation is opening of the blood vessel, constriction is the closing, so we can have control of blood pressure. They're involved in clotting, and they're also very, very important in inflammation. They can be pro-inflammatory or anti-inflammatory, depending on the nature of the molecule that's generated. And so if you've got a headache, as an example, sometimes that can be due to vasoconstriction. You'll take aspirin. Aspirin mediates its effect because it actually blocks the enzyme that converts arachidonic acid into prostaglandins, so the cyclooxygenase. And as a result, because we get all of these products, we get wide biological effects from the use of just aspirin, because we're knocking off so many different molecules that are generated by this precursor. So if we look here, this is the metabolism of arachidonic acid into other biologically active molecules. So we start off with arachidonate, just another way to refer to arachidonic acid. Phospholipids through phospholipase A2 can generate arachidonic acid. We can have a diacyl glycerol. So this is a fatty acid or a glycerol molecule with just 2 fatty acids. That might be a phospholipid. Through another lipase, it can be generated. And once we've got this molecule, this is at the center, it's metabolized. So if it's metabolized by the lipoxygenase, you end up with your leukotrienes. If it's through the prostaglandins, you generate your key prostaglandins, and we've got them here, prostaglandin H2, and then that is then metabolized into other things like prostacyclins, other prostaglandins, and thromboxanes, of which there are many. So you can see that there's a massive amount of different molecules that are generated, and they all can have those biological activities that we saw right up here. So remember, if we block cyclooxygenase, this is prostaglandin, but if we say we're blocking the cyclooxygenase with aspirin, we're going to be blocking all of these activities and these products. So that brings me to the end of this lecture. So again, a reasonable amount, but I think it's all fairly simple and straightforward. We've got the summary, which highlights a lot of those key points, and you've also got your tutorial questions. So make sure you can do the beta oxidation calculation. It's even part of your tutorial because that's important, and you have a broad understanding of the roles of fatty acids and also how the glycerol molecule is used within the cell. Okay, see you in the next record. Okay, welcome to the next lecture recording. So this lecture we're going to talk principally about cholesterol and then some of the byproducts that give rise to steroids and hormones. So this will really answer this question about why do we need cholesterol within our body. So we're going to talk about the biological roles of cholesterol within the host, the 3 steps that are used to synthesize cholesterol. We'll talk about the importance of this molecule, HMG-CoA reductase, or this is an enzyme, and we spoke about HMG in the previous lecture. We'll also talk about how cholesterol is regulated, and again, this feeds into the reductase enzyme. We'll talk about the transportation of cholesterol in the blood, and also the biological functions of cholesterol or other biological functions. So again, space for comments and questions. So we know that cholesterol has received a lot of attention due to its link with heart disease, but what people may not appreciate is that cholesterol is actually essential for life. You must have it. It's actually a component of all cell membranes, and by doing so, it actually regulates the fluidity of the membrane. We saw this with fatty acids previously. It's also the precursor for all steroid hormones, so testosterone and cortisol as 2 examples, and these are just essential for life. So you can see why this is going to be the case. This is the structure of cholesterol. So you can see there are 27 carbon atoms that are generated. You don't need to know the structure or draw the structure, but we'll show the synthesis in the latter portion of this lecture. So what are the sources of cholesterol and what about its synthesis? So typical diet will have ample amounts of cholesterol in our diet, so we don't actually need to synthesize it. So again, this is part of the logic that if it's already present, there's no point for the body expending energy and resources to make something. But we do know that it can also be synthesized, and this is done predominantly in the liver. Now, where do those 27 carbon atoms come from? They all come from acetyl-CoA. And this is very much going to link in to the previous lecture. And we'll also start to talk about ketone bodies again, but in a slightly different context. So make sure you cover this lecture and the previous lecture together because they are very much interconnected. So there are 3 steps in generating cholesterol. And we'll go through them briefly and then we'll go into the detail below. So the first one is the synthesis of isopentyl pyrophosphate. And this is something that occurs in the cytoplasm. You've then got that term again of condensation. So this is where you've got the isopentyl pyrophosphate being added to itself to form squalene. This again still occurs in the cytoplasm. And then the squalene starts to cyclize, and you've got these circular ring-like structures above. And this is what will form cholesterol. And this occurs in the endoplasmic reticulum of the liver cell, of the hepatocyte. So the first step. So 3-hydroxy 3-methylglycerol-CoA. We spoke about this previously, and I said you don't worry about the name, but remember the abbreviation, HMG-CoA. So here we are again. So again, an important molecule for fatty acids, but also, and in that context, we're talking about it with ketone bodies, but now we're going to talk about it in relation to cholesterol. This is formed from acetyl-CoA and acetoacetyl-CoA. Remember, acetyl-CoA, when you've got 2 of them, forms acetoacetyl-CoA. So this is the same process as the formation of ketone bodies, as I mentioned, see the lipids lecture. Now, the location is important for the different reactions. So, in the cytosol, HMG-CoA is converted to mevanolate, while in the mitochondria, it leads to acetyl-CoA and acetoacetate. So what we see here is that depending on the movement of HMG-CoA, we either generate mevanolate, which is a precursor of cholesterol, or we form ketone bodies. And this is what the diagram below is showing. Now, this part here, we sort of had part of this in the previous recording, but certainly we spoke more about this latter portion. So here is acetyl-CoA, acetoacetyl-CoA, which remember, as I mentioned before, acetyl-CoA times 2 generates this. So 2 acetyl-CoAs gives you acetoacetyl-CoA. You add another unit, and we end up with our HMG-CoA. Depending on the location, so cytosol versus the mitochondria, you end up with mevanolate, and this is now going down the cholesterol pathway, or acetyl-CoA and acetoacetate, which is now going down the ketone pathway. So this one precursor can go down either pathway depending on the requirements of the cell, and therefore the host. So if there is a requirement for the cholesterol, you're going to get more of it going in this direction. If energy is required, it's going to go down this pathway. So as it shows you here, 2 pathways where HMG-CoA can lead to the synthesis of ketones or cholesterol. And so HMG-CoA is reduced to form mevanolate, and so you can see here you've got NADPH, it's reduced, gives rise to NADP plus through the HMG-CoA reductase. Because this is the first committed step, so first committed step, this is also going to be the rate limiting step of this process, because as this goes on, this is what is going to give rise to our cholesterol synthesis. So you can see this breakdown of this molecule. I've given you the same idea here. Synthesis of malate is the committed step in the synthesis of cholesterol, so therefore the HMG-CoA reductase is an important control site for cholesterol synthesis. This again is another example where we're going through a pathway. We talk about the steps of the pathway, but we also talk about the control points. So hopefully you're starting to see a pattern. This is going to be another topic or another section where we're going to highlight these as important molecules. So the important molecule in this case is going to be the enzyme HMG-CoA reductase. As we said in the previous step, it's basically an addition reaction. Mevanolate is converted into 3-isopentyl pyrophosphate. You can see that here. Don't worry too much about the chemistry. Just look at the basic process. So mevanolate, you've got ATP being used. So it's an energy-intensive process. 5-phosphomevanolate. So this is the phosphate group that was attached. You repeat the process again. 5-pyrophosphate mevanolate. This is the second group. Remember, pyrophosphate is just 2 phosphates. Again, more energy is involved, but it's now in a different location. And then we lose one of those phosphates. Why do we lose it? Because it makes it a point of reactivity here to then generate this product, which is our 3-isopentyl pyrophosphate. This molecule then continues and basically just gets added onto itself. And that's what step 2 is showing you here. 3-isopentyl pyrophosphate. That molecule there condenses to form squalene. So this is 3, 4, 5. Oh, sorry, T1, 2, 3, 4, and 5 carbon atoms. Is the carbon atom. We add 2 together, we make a 10, then a 15, and then 2 of those 15 units come together to make a 30. And remember that cholesterol was this 27 carbon atom molecule. So here we go. This is just showing you those groups. The pyrophosphate, isopentyl pyrophosphate comes together, generates an intermediate. Another isopentyl phosphate comes together, generates the next intermediate, and this ultimately gives rise to the squalene. Do you need to worry about this one, or this one? No. You just need to know that it is basically a repeat process of this precursor, and it's just an addition to give rise to squalene. Squalene, as you can see, as it folds together, is going to give rise to your cholesterol molecule, and that's what this is showing you here, step 3. Squalene folds onto itself through a process of cyclization to form cholesterol. Again, it's showing you all of the steps. Do you need to know all of these steps? No. It's just showing you how does squalene make its way down to cholesterol. It's showing you where the bonds form and the formation of the 5 and 6 membered ring structures. And you see, there's actually a lot of steps, but we're not going to go through all of those steps. It's just this gives rise to cholesterol, and this is the important molecule. Okay, so I'll finish off just on this last one here, so cholesterol regulation. So we ingest excess cholesterol from our diet. As a result, how does the body regulate the cholesterol synthesis? It's actually very simple. The amount and activity of the HMG-CoA reductase, so the amount refers to the amount of protein, its activity refers to the amount of reactions it's able to catalyze, is the key control point. Why? Because remember, this is the first committed step, and it is also the rate limiting step. So this is the one that we want to focus on. And there are 4 approaches. One is the rate of synthesis of the enzyme itself at the messenger RNA level. If you reduce the level of message, you're going to ultimately reduce the level of protein, and we'll see this in subsequent lectures. mRNA levels can also be inhibited by mevanolate and its metabolite, so this is another example of negative feedback. We can degrade the HMG-CoA reductase itself by destroying it, then it's not going to allow it to convert. And we can also phosphorylate the HMG-CoA reductase to turn off its activity. So you see there's multiple ways that its activity can be regulated within the host. Why would you want to have multiple points? Because this is such an important molecule. It has so many different downstream functions, you need to have multiple ways of being able to regulate its activity. Okay, I'll stop there. We'll move on to different aspects of cholesterol in the next recording. Okay, welcome back. So now let's move on to talk about cholesterol, but more importantly, its transport. So we know that cholesterol is made in the liver because that's all that we spoke about above, but it's used throughout the body because it's part of the cell membrane. So again, how is cholesterol transported in the blood? Cholesterol and triacylglycerol, so remember fatty acids, are packaged in lipoprotein, so it's the same idea. We've got albumin, or protein carriers, that are involved in transporting these molecules. So when we look at a lipoprotein, so remember it's going to be a complex between lipids, and the lipids could be triacylglycerol or cholesterol, and proteins. Each one of these particles has a core of hydrophobic lipids, which we know that lipids are going to be hydrophobic by their very nature, surrounded by more polar lipids and proteins. So lipids that are polar are going to want to exist in an aqueous environment, and certainly proteins are going to have components that are going to be hydrophilic. The proteins though, the protein components, which are also referred to as apoproteins, are important in solubilization. So they make these hydrophobic lipids more soluble, but also they allow for cellular targeting. It is how the cell is able to take up the cholesterol. It binds to the protein. By binding to the protein, the lipids come along for the ride, and that allows the protein to bring all of these other lipids into the particular cell. So if that cell needs carbohydrate, it wouldn't, so that's a mistake. If it needs cholesterol, apologies, it would bind to that protein and bring in cholesterol into the cell. Now, these lipoproteins are all classified according to their density that will increase, and you'll have all heard of these multiple times. We have low density lipoproteins, or LDL, and this is the major carrier of cholesterol in the blood, and it's referred to as the bad cholesterol, if you want to keep it as good and bad. LDL transports cholesterol to peripheral tissues, and it also regulates the de novo synthesis at those sites. So this is the synthesis of the cholesterol. The other one is the high density lipoproteins, or HDL. And so when you go to the doctor and they'll measure your cholesterol, they will always measure your LDL and your HDL, and it's the ratio between the two. HDL is considered the good cholesterol because it collects cholesterol in the plasma that's released from dying cells. And as a result, it can be thought of as mopping up the cholesterol. So when you've got more HDL, it provides those protective effects. And cholesterol can also be taken up by cells through receptor-mediated endocytosis. The receptor is going to be binding to those proteins, and that endocytosis is just the internalization of that lipid particle into the cell. So this is a cross-section of one of those lipids. So you can see here cholesterol, ester in yellow, the bulk of the cholesterol is in the middle. You've got a protein, apoprotein, shown in blue. You've got unesterified cholesterol, which is scattered throughout, and you've got predominantly phospholipids, which is on the outside. And remember, phospholipid is part of the cell membrane. So not surprisingly, it's going to be on the outside of the lipoprotein. It's going to be stable there. Here are the different examples of the sizes. So you've got chylomicrons, these are the least dense. Then you've got very low density, you've got intermediate, low density, and high density lipoproteins. And you can see the density going up and up and up as we go through. And you can see that the size is conversely going smaller and smaller. So the packaging is getting more dense. There are different lipoproteins on the surface, and you can see their main functions. Don't worry about all of these details, but just focus on these last two, where we've got the different proteins and really their functions. So transport and reverse transport of cholesterol. We're not going to worry too much about those other molecules. So this diagram here is showing you the steps involved in the internalization of cholesterol into the cell. So we have low density lipoprotein, you've got cholesterol in the center, and you've got protein on the outside. So this is showing you the same things in this diagram. So here's the protein in blue, protein, and your cholesterol is either there or it's on the inside. Okay. Now, on the surface of the cell, you'll have LDL receptors. These receptors are going to be specific to the protein. So as the protein comes closer to the cell, if those receptors are expressed, you're going to get binding. It binds, it is internalized. Those receptors are then removed and go back to the surface of the cell. So we've got a recycling event. The lipids are then broken up. So we've got amino acids from the protein. Remember, this is the receptor. So this is what is linked with the carrier protein. You've got the cholesterol that has been released. This is then converted into cholesterol, and it's used by the cell either to go into the cell membrane, or as we will see later, to make steroids and other biological molecules within the cell. So let's talk about steroid synthesis, because that really follows on very nicely from the previous point. Cholesterol is a precursor for all bile salts, steroid hormones, and vitamin D. So vitamin D is important for bone health. Steroids are important for pretty much all of the functions within the body, some of the key characteristics such as gender, and bile salts are important for digestion. So as it says here, bile salts act as a detergent, helping to digest or break down ingested lipids. Cholesterol leads to the formation of steroids. So if we look at progesterone, this is important for preparing the uterus for implantation. You've got androgens, important for male sex characteristics such as testosterone, estrogen for female characteristics such as estrogen, and glucocorticoids that give rise to gluconeogenesis or promote gluconeogenesis such as cortisol, and mineral corticoids that affect blood pressure such as aldosterone. So if you look at these are major things within the host, major things within the body. So again, you can tell that